completed the 4 lecture sessions, which were virtual from US, this Malaysia, Korea, and one more was there. So, in continuation of the scientific lectures, we are organizing this virtual oration competition. So let me brief about the things here. This virtual oration competition will lead to 4 Young Researcher Awards, and competitions are happening in 3 parts. You will be each participant will be evaluated out of 10 marks from the each one, and we will take a average score, and those who will be at the top 4 position, they will get the 4 Young Researcher Award, which are attached to in the memory of some deceased, and they all are sponsored award. We have attached the cash or check money with them. Yeah, I have already mentioned the details in the oral competition schedule. Uh, Let me introduce about our judges. So first competition will be led by Professor Gaurav Gupta, who is adjunct research professor at Center of Medical and Bioallied Health Sciences Research, Ajman University in United Arab Emirates. And that session will be supported by Dr. Simon Walcott, who is Chief Technician at Department of Chemistry at FST, University of West Indies, Trinidad and Tobago. Second session will be judged by Professor Sadish Kumar, who is Director of ITS College of Pharmacy, Muradnagar, Ghaziabad, Uttar Pradesh, and it will be supported by Professor Vithal Chopde, Department of Pharmaceutical Chemistry, PES, Modern College of Pharmacy, Nigdi, Pune, Maharashtra. Third session, which will occur in 3 links of 40 minutes, each having 3 and 2 presentations, will be judged by Dr. Saurabh Daya, who is Dean of Administration and Student Affairs, Delhi Pharmaceutical Sciences and Research University. And it will be, unfortunately, Professor Jyoti and Professor Tapan. I don't know whether they are here or not, but we have the support of Professor Mehul Patel, who will join as the co-judge with the Dr. Saurabh Daya for the scoring. So this was briefing about our virtual oration competition, and here we are commemorating the World AIDS Day, which happened on the 1st December. Always we are attaching our APP conferences to some of the health days so that we can also support the initiative of World Health Organization. Taking no more time, I ask Professor Mittal is not there. In that case, we have Professor P. Shanmugasundram, who is our president of APP Tamil Nadu State Branch and Dean of School of Pharmaceutical Sciences at Wells University, for 5-minute inaugural remarks. Professor Shanmugasundram, please.
Yeah, thank you, Professor Dagia, sir. It's a great honor to be a part of this 14th annual convention of APP, and we are continuously for more than associated with this association for more than 10 years from Vistas. And this year also our students, business scholars are participating, and one of our faculty is getting some awards in a validation. And Dr. Karthikeyan from Vistas is a speaker, offline speaker. Tomorrow he'll be in Vistas. Thanks for the opportunities provided to the young talents by Dahia sir and the association for promoting all these talents. And from the morning, it was I was in the inaugural session, and it was great. 4 scientific lectures from Korea, Malaysia, and from other personality. So all this. That was a forenoon session was very good, and now I hope the competitive and the afternoon session also will be a interesting one. It paves way to all the youngsters to produce the talent. Thank you, Daya sir, for this wonderful opportunity. Thanks for continuously supporting the younger generation. Thank you once again.
Thank you, Professor Shanmugasundaram, for your inaugural remarks. Now I invite Professor Deepak Kumar, who is head of our APP Solon local branch and presently Dean of School of Pharmaceutical Sciences at Sulini University Solon for the inaugural remarks. Dr. Deepak.
Hello, good afternoon, sir.
Yes, you
Yes, so
can
sorry,
deliver
sir.
your remark.
I was delayed, sir, because I was in the meeting, sir, and again I need to go for the meeting, sir. Yes, sir.
You can go ahead for your inaugural remarks.
Okay, sir. Okay. I'm Dr. Deepak Kumar, and I'm the professor and dean School of Pharmaceutical Sciences. And we are, or with the help of APP, we are purposing to organize one international conference here in Sulni University also. For this, we need the support of Rajiv Dahiya sir, and and recently many of our students are also participating in the conferences organized by the APP, and they have also been the members of APP, and we want again our association with the APP. Thank
Okay,
you, sir.
thank you.
Yes.
Uh, okay, so we have Professor Mehul Patel, who is our former president of APP Gujarat State Branch, and he's a professor of pharmaceutical technology at Dharam Singh Desai University, Nadiad. So, Professor Patel, if you want to address us.
A very good afternoon, all the participants and resource persons that gather here for this virtual oration competitions that is organized by APP branches of Andhra Pradesh and the molecular pharmacology divisions and the host institute, Vijay Institute of Pharmaceutical Sciences for Women, Vijayawada, Andhra Pradesh. So, as already Sir has inaugurated that sessions where Sir has highlighted that the research scholars, this kind of session definitely benefits all the research scholars and research buddies, those who want to explore the more and more area in the field of in the various field of the pharmacy. And this kind of platforms share the knowledge between the students or among the community of the communities. Even we are now getting benefits by this platform from the foreign research scholars can also join with our students. So even the research students from the Indians also come to know that what kind of different research that going out throughout the world. So definitely this oration competitions that helps to the participants as well as well the delegates or audience that are looking want to pursue the such kind of research. So thank you once again to the APP and host institutions for giving this opportunity. Thank you very much.
Thank you, Professor Patel. We have another personality, Professor Gupta, Gaurav Gupta, who is our president of APP Gulf International Branch and is associated with the Ajman University. Professor Gupta, if you are with us, you can share your views.
Yeah, a very good afternoon to all of you, and welcome to virtual oral competition to recognize a outstanding research skills. As I am seeing that uh, whenever Dr. Dahiya organizing any conferences, they are associating this conference and organizing these conferences on some particular special days. Like now, today is a World AIDS Day. So it is very good thing that they are organizing a conferences on some particular disease day so that we are remembering those type of diseases and we are getting aware of those types of diseases. Otherwise, if we are seeing that we are not celebrating, we are not seeing any awareness of this AIDS day anywhere in the society. So this is a good platform to arrange or to organize a conference as well as to memorize the particular disease on a particular day. So this is a good platform. As well as this is a best platform for the young generations to improve their communication skills, to improve their giving a presentation skills, and so that is the best platform. So I would expect to each and every principal of the institutes who are there, who are joining with us, so that they can aware their students to join as much as possible in this type of competitions, so that they can also get a chance to give a talk and they can also get the prize. So wish you a good luck to all the speakers and as well as the presenters for this conference and for these competitions. Thank you.
Thank you, Professor Gupta, for your kind words. We have another personality, Dr. Walcott, who is Vice President of our APP West Indies International Branch and is associated with Faculty of Science and Technology, University of the West Indies at Trinidad and Tobago. Dr. Walcott, if you want to share some views. Please unmute
Yes,
yourself.
thank you. Yes, thank you for that. It's really my honor to join you all for the international convention. I enjoyed some of the presentations a while ago. It's really eye-opening to hear what's on the cutting edge of research. And I really did notice that we have a lot of these conferences happening around the day, and I understand why now. So it's targeted to various health days and health issues that we're having, which affects us all around the globe. So today's gathering is especially meaningful as it is held in commemoration, as was said a while ago, of World AIDS Day. This reminds us of both the extraordinary progress that science has made and the continued responsibility we bear as researchers and healthcare professionals. It calls us to pursue discovery not just for intellectual pursuits, but also with compassion and purpose. So I am really happy to be here. I'm looking forward to the presentations. Good luck to everyone. I'm looking forward to hearing about the work done in terms of pharmacology, medicinal chemistry, nano research. So I must commend our host as well for having thought about this at this particular time. So I am looking forward to the presentations. Thank you, Prof. Dahia, for consideration as well.
Thank you, Dr. Walcott. We have another judge today, Professor Chopde, who is professor of chemistry from Pune. Professor Chopde, if you want to share some views, we have some time.
Yeah, sir. Thank you, sir, for giving me a good opportunity to spare the valuable time with you. So first of all, I'm very much thankful to Dr. Dahiya, sir, who is a very dynamic and extraordinary personality. Is with us because he's very good and interested to conduct the different type of research as well as the gathering of the all pharmacy peoples at one place, and that is the need of today's, because we all researchers are working in our different places, but we don't know what exactly is happening. And Dr. Daya sir is taking the efforts to gather all researcher at one place. They organize the different conferences. He's taking the very much interested to organize the research presentations for the young researchers, and he give the motivational awards to them. Due to that motivations, people can do the good and extraordinary research, which will be really valuable for the today's scenario. Because we know that just few years back we had faced some pandemic situations. So that it clearly indicates that all researchers should be very much ready, and they should be always ready to develop the different type of the research which are needful for the society. So I'm with this Association of Pharmacy. I'm associated with this APP from the last 2-3 years, and I have seen a lot of efforts like Gupta sirs and other so many dignitaries are there from different countries, not only in India, in different countries, and that is a clearly indicate that we are getting the good opportunity to connect the people who are working in the research in the worldwide, not only in the one country. So all credits are goes to the all team, and most of the team leaders, Dr. Raju sir is with us. So I'm very much thankful to Raju sirs and other teams who have given me the good opportunity to work as a chairman for some sessions on the occasion of this 14th annual conventions of the APP. So surely that the session will be helpful to all researcher, not only in India, but that will be from in the different all other countries also. So all those are also I'm appealing to all those are associated with this association. They should actively participate the step up the conventions, and they should organize this type of convention physically as well as virtually in the hybrid mode. So it will be helpful to the other peoples, and people should know that what the APP is doing. And so many associations are there. They are arranging the different conferences and other the scientific compositions on their own places at that level based. So our APP should be come like in that page. In India, in the 19th December, All India Pharmaceutical Congress is there. There is the IPC, Indian Pharmaceutical Congress. More than 10,000 delegates are participating that conference. So that is limited for India for participation. They also allowed for the abroad also, but very limited participants are getting from the abroad. But as far as our connectivity and the assets are concerned, we can call them as assets. We have all what people are joined with us. That is our assets. We can call that our intellectual assets. Like just Madam was told that she is from the other countries and like the USA, Germany, Europe, okay, and Korea and other country. Very active our peoples are there. They are our not treasure assets. So we can use that type of our assets, and we should always conduct this type of annual conventions as well as the conferences at your places. Now the country-wise here is responsibility to give the much more registration to their who will get some financial help also, and we are getting the good gathering, and that gathering will be helpful for the developing the country as well as the research developing the pharmaceutical field.
Okay, thank you, Prof.
Lastly, I'm thankful to Raju sir for the giving the opportunity.
So
thank you,
thank you,
very much,
Professor. Yeah. So, Jansiana, Dr. Jansiana, have you got response from your supervisor? You please unmute.
Yes, she cannot come. I'm very sorry.
Okay, no problem, no problem. So we have another president here, president of Karnataka State Branch, Professor Kuntal Das, who is associated with the Balai College of Pharmacy. Professor Das.
Yeah, good afternoon, sir.
Yeah, you can share your views.
So this Association of Pharmacy Professionals APP under the leadership of Dr. Rajiv Daya sir. So we are improving every angle along with the pharmacy student, PharmD student, not only the pharmacy student, we are getting the overall countries several opportunities, and sir has given the scientific platform where students also going for participation and they're getting the awards, all the recognitions and all. Even we are also getting the lot of opportunities with the APP, and we are gathering a lot of people and we are performing well, and in future also we are thinking we are getting the lot of opportunities given by him. Thank you, sir.
Thank you, Professor Das. Professor Padmalatha, you are here? Professor Padmalatha.
Yes, sir.
Yeah. So we have our hosting principal as well as president of APP Andhra State Branch to share her views. Madam, please.
Sir, we are very much honored to address the gathering, and I'm very happy to associate with the APP, the national and international branches, and this has given us a very good platform to interact with the foreign researchers and speakers. And regarding today's program, if you look into all the talks, all the lectures are very, very informative for the student level. When students are able to follow the each and every topic, so I'm very happy for hosting such a wonderful program. And thanks to Raju Dahiya and Dr. Sunita Dahiya, madam. And will continue the relations further and for the betterment of the students.
Sir,
And
join
the
कर लो,
students, this is
wait
a
कर लो.
platform the students are given.
Okay, okay, okay. Thanks.
This is a platform where students are also appreciated and given best student awards for their academic excellence, even in their research areas. So thanks to Dr. Raju Dahiya for his thoughts and promoting the student community. Thank you, sir.
Thank you, Padmalata madam. Thank you for your support also. Without
Thank
your support, I would not be able to do much in the southern part of India. So you and Professor Shanmuga Sundaram are supporting a lot. And I'm very happy to honor your students within the best student category. So
well,
I'm
thank
just,
you.
yeah, thank you. I'm just waiting on one of our more president. Oh, we have our one of the another colleague, Professor Satish Kumar. Yes, Professor Satish Kumar.
Yeah.
Yeah,
Can you hear
so
me?
yes, Professor Satish Kumar is the director of ITS College of Pharmacy, Ghaziabad, and is one of the chairperson today. You can share your views, sir.
So can you hear me?
Yeah, you can share your views. Everybody is hearing you.
Right. So I'm extremely sorry that I could not join in time. So good afternoon, one and all. Good afternoon. Yeah. Uh, yeah. Really, it gives me a great pleasure to be a part of this conference, and I really thank Dr. Rajiv Daya, President APP, is doing a wonderful job connecting the whole world, through this platform as well. And all the very best to the presenters. And I'll be there for chairing a session as well a short while from now. And thank you very much, Dr. Daya. Thank you.
Yeah, thank you, Professor Sadish Kumar. Now we have our last inaugural remarks, which will be from Dr. Kamal Dua, and he is president of our Australian International Branch and attached to the University of Technology, Sydney. Dr. Dua, please.
Thank you. Thank you so much, Dr. Daya. And first of all, my apologies for joining a bit late because I was in a different meeting and I got mixed up with the time. So I have sent my apologies to Dr. Daya, but thank you so much for having me. And a very good afternoon, actually. I believe it should be there, every one of you distinguished colleagues, honored guests, and friends. And I would really like to welcome to this conference of the Association of Pharmacy Professional, and Dr. Daya had chosen me to give the inaugural remark as being the president for the Australian branch in collaboration with all these international different partners of the association. I would really like to welcome you all for the conference, and it is absolutely a pleasure for me actually to deliver this inaugural remarks. And thanks to Dr. Daya again for inviting me for this. First of all, I would like to acknowledge the commitment actually and the passion for each one of you actually, whether you are a practitioner, a pharmacist, or researcher, student, or a teacher, an educator, which all brings us together to this profession together. And as we know that it is quite a rapid, time after COVID, which has significantly evolved over the period of time in the healthcare system, the pharmaceutical sciences, regulations, and also the public expectations. Particularly if I talk about in Australia, the role of pharmacist and is completely changing. We have something called at the moment in practice called as an expanded scope of pharmacy. And I believe that's the same thing happening in the other part of the world as well. When we talk about the expanded scope of pharmacy, where the pharmacists are now being allowed even to prescribe the medications for certain elements, In NSW in New South Wales in Australia, we have certain trials even runnings for UTI and certain skin disorders. And these kind of the conferences, particularly your presence, actually clearly shows a testament of your dedication, which are very much towards the patient-centered. Now, as this conference, as I mentioned, that it is really not only a platform which unites us across the different continents, the people, particularly it's happening in India at the moment, but across the different disciplines. And it helps us to actually share our insights and also to learn from the learned speakers, those which are in the lobby of this conference across the 2 days. APP really had a very long history and it's almost more than a decade. And I know the passion and also the hard work of Dr. Daya as a president. I've been definitely doing a little bit parts, but I believe the fueling of this association is particularly through Dr. Daya. It's a long history where we all come together of the pharmaceutical scientists, educators, practitioners, and even industries. And these type of the conferences and the association help us actually fostering the collaboration and also promoting the best practice, which is the one of the main stigma and the main, the thing which is going around the globe to help these kind of the bodies bringing it up. Now, over the 2 days, today and tomorrow, you already been learning from different speakers, and I'm sure you'll have an opportunity to explore and emerging to see the different trends and the emerging opportunities in the area of drug discovery, development, and in clinical practice, and moreover in pharmaceutical education. Because I can clearly see the different speakers from different countries which have been speaking in this. The ultimate goal, which I personally feel for all of us through this conference, is to improve the healthcare outcomes and to enhance the patient care through learning and emerging and embedding into the new research areas. Because the globe is completely changing and the evolution is happening every day in every form, in every discipline. And particularly with this hope, I can say that this conference is helping us to reconnect and to share and to commit on something we all can work together. I'm sure there are a lot of people on this screen which I know personally and professionally. And it is particularly an opportunity for the young researchers, particularly, I should say, as an ECRs, emerging career researchers, where you can not only just meet the other senior leaders, but also to connect with themselves. I'm not going to take too much of time. I think I'm speaking a lot and I do speak a lot, so I really need to pause myself. But I encourage that all of you to take the advantage of this gathering and a conference and to engage yourself in different discussions, to challenge certain ideas, to ask some difficult questions, and to explore the new possibilities. We should definitely use this conference not only to just reflect what we got, but to learn something where we want to go, to advance the pharmacy profession as a vibrant, I should say, or a responsive, or I should say scientifically rigorous, and all above, I should say, as a socially responsible profession. And on behalf of the Australian branch of Association of Pharmacy Professional and being a president, I would like to extend the warmest welcome to you all to this conference. And thank you for inviting me to being for the opening remarks. And let us all make this conference a milestone collectively in our journey forward. With all these things, thank you so much again for being patiently listening to me. And thanks, Dr. Daya, for being the president for the APP, for inviting me and giving this opportunity to talk. Thank you.
Thank you, Dr. Dua, for your remarks. So now I invite all the judges, especially Professor Gaurav Gupta, to lead through link 1 and start the presentation, and Professor Sadish Kumar through link 2, and Dr. Saurabh Dhaya and Mehul Patel, sir, for link 3. Thank you. If any difficulty is coming, you can call me on via WhatsApp.
These are mostly is widely caused fungal cases. So we have to
Yeah, hello everyone. Yeah, hi Camelia. Is Camelia is here?
Yes, I can hear you now.
Yeah, Camelia, shall we start now?
Yes.
Okay, please, you can now start your presentation. And please note it time. It's you have a 10 minutes of time for the presentation. And next, then 2 to 3 minutes will be follow for the discussion.
Yes. Thank you.
Yeah, please.
Good morning, good afternoon, good evening, everyone. My name is Gentiana Chubuk George, and I'm a first year PhD student at Western University, Arad, Romania. Today, I'll present my review on homeopathy, reconceptualized as an emerging form of nanomedicine. This work synthesizes 2 decades of physicochemical and biological evidence that challenges traditional views about ultra-high dilutions. Homeopathy has been controversial for over 200 years because of this fundamental paradox. Remedies are diluted far beyond Avogadro's limit, sometimes 10 to the power of 400, where statistically no molecules of the original substance should remain. Critics argue these preparations must be pharmacologically inert, yet homeopathy
sorry to interrupt you.
persists globally with substantial clinical use and patient-reported benefits. This disconnect between theory and practice has motivated renewed scientific investigation.
Camelia, your slides are not visible.
Oh,
Yeah, could you please make it visible?
yes, okay. Is it
Yes.
okay now?
Yes, yes, it's visible.
Recent advances in nanotechnology offer a potential explanation. Multiple research groups using sophisticated analytical techniques, electron microscopy, light scattering, mass spectrometry, have detected nanoparticles in potentized preparations. These nanostructures typically range from 1 to 100 nanometers and appear to originate from the starting material, stabilized by organic coatings. This raises a provocative question. Could homeopathy represent a nearly empirical form of nanomedicine? The physicochemical evidence is compelling. Chikramani and colleagues demonstrated that metal remedies contain nanoparticles even at extreme dilutions like 200 C. These particles measure 5 to 15 nanometers, and their concentration stabilizes after the 6 C dilution. Van Wassenhoven's team confirmed these findings using multiple analytical techniques. The mechanical energy from succussion, the vigorous shaking process, appears to generate nanoparticles through cavitation and container wall abrasion, creating stable colloidal systems. Biological studies reveal activity patterns characteristic of nanopharmacology. In vitro work shows nonlinear hormetic dose-response curves, where low doses trigger beneficial adaptive responses. Chikramani demonstrated that metal nanoparticles at picogram per milliliter concentrations elicited significant biological responses in yeast stress models. In vivo, Kudabuksh reported that remedies diluted beyond Avogadro's limit modulated tumor suppressor proteins like P53 and BCL2. These multi-target system-level effects resemble complex nanocarriers rather than conventional drugs. When we compare homeopathic preparations with established nanomedicines, striking parallels emerge. Both operate in the 1 to 100 nanometer range, show high surface reactivity, cross biological barriers, and produce effects at extremely low concentrations. Bell and Coyden's NPCAS model proposes that remedy nanoparticles act as mild biological stressors, triggering hormetic adaptive responses through network-level mechanisms. This framework integrates stress biology, allostasis, and nanopharmacology into a testable hypothesis. Despite promising findings, significant limitations remain. Many studies use small samples with inadequate controls, making it hard to distinguish genuine nanostructures from contamination. The field lacks standardized protocols, reference materials, and interlaboratory validation. A few investigations integrate detailed physicochemical characterization with functional assays on the same preparations. Most research focuses on individual remedies, so generalizability across different manufacturers and clinical applications remains uncertain. These methodological gaps must be addressed before definitive conclusions can be drawn. Moving forward, several priorities emerge. First, the field urgently needs internationally agreed protocols for preparation, characterization, and quality control, with validated analytical methods and interlaboratory testing. Second, mechanistic studies must integrate nanoparticle profiling with functional assays using rigorous experimental design. Third, large-scale collaborative networks should prioritize independent replication with transparent data sharing. Finally, translational research should explore whether nanoscale characterization can predict clinical outcomes, enabling rational formulation design. Interdisciplinary dialogue among practitioners, nanoscientists, pharmacologists, and regulators will be essential. To summarize, 2 decades of research provide accumulating evidence that homeopathic preparations contain nanoparticles and exhibit biological activity consistent with nanopharmacology. This represents a potential paradigm shift, reframing homeopathy not as pre-scientific, but as an inadvertent precursor to modern colloidal nanopharmaceuticals. However, critical methodological limitations remain. Standardization, rigorous replication, and transparent research are essential. Whether this reconceptualization proves valid remains to be determined. But if validated through high-quality studies, it could open novel avenues for precision, low-dose therapeutic interventions. In conclusion, the nanomedicine hypothesis offers a scientifically grounded framework for understanding homeopathy. It bridges 2 centuries of traditional practice with cutting-edge nanoscience, providing testable hypotheses and clear research priorities. The evidence is preliminary but provocative, requiring rigorous validation through interdisciplinary collaboration. If confirmed, this paradigm shift has profound implications. For nanomedicine, it offers new insights into low-dose mechanisms. For homeopathy, it provides a rational foundation for observed effects. And for pharmaceutical practice, it suggests pathways for integrating traditional and modern approaches. As one researcher noted, The question is no longer whether something is present in these preparations, but what it is and how it works. I wish to thank Dr. Rajan Sankaran, whose elevation of homeopathic practice to an art form inspired me to explore its scientific foundations. I am deeply grateful to my teacher, academician Professor Dumitru Dobrescu, whose pioneering work on homeopathic pharmacology and introduction of the nanopharmacology concept in 2007 provided the conceptual foundation for this review. Full references are in the manuscript. Thank you for your attention.
Thanks a lot, Camelia, for a informative presentation. Now, I would like to request to Chairperson Dr. Simon for his comment on your presentation. Dr. Simon, do you have any queries?
I would like to ask one small question. Can I proceed?
Yes, sure.
Yeah, sure. I appreciate your interest in the medicine, especially the traditional medicine and nanomedicine together. So I greatly appreciate first because you are also looking for homeopathic medicine together with nanomedicine, right?
Yes.
Yeah, great. So actually, I've been very fond of the Ayurveda, but I never had chance to know even basic concept of the homeopathy. But there was some rumors as well. So because of rumors, there was never been passion to look after the because of once a rumor it becomes some negativity in the homeopathy, like they use some kind of heavy metals together and then that kind of I think it's like not fit in the my idea of safety and all those. So please give some insight about the are there concern about like medicine itself, some kind of health hazards, like dose, if there is excessive dose, and if there are some like some historic, there's some example like in the patient had some
Professor, your voice is very disturbed. If you can repeat the question.
yeah, I will repeat just a minute. I would like to know about how we can make the popularity and address the dilemma about the homeopathy among the patient. Like they have some kind of the pre the, they have the misconceptions about the homeopathy. Some of the people. So your long experience about the homeopathy. Is there any concern of overdose or toxicity or great concern? Because I want to be also come into the enter into the understanding about the homeopathic, and I also work in nanomedicine. We will look after that session, sharing the Google Scholar. We work for the cancer, multi-drug resistance, especially for the diabetic wound healing, and some sort of the uh, some new age medicine, because of the multi-drug resistance is going to be 100 times plus crisis than the COVID-19 if it's not properly addressed, because it's already killing the critical patients, the multi-drug resistant pathogens, and they are not one, they are n number of pathogens are there. So we have to also took the focus getting worse because of the pollution of the antibiotics, the speed of the emergent superbugs is just like a very vertical. I would say it's like it is vertical. So they are, and I personally believe that nanomedicine can be the only solution because there are a number of reasons. And bacteria is a super genius, intelligence, can deal with many things. But we have to consider their mechanisms, all sort of mechanisms, and target the specificity that will be no more possibility of developing resistance to our medicine. So total break. There's this like make the history, like era. Multi-drug resistance era was before, but no more. So that vision which we bring and see the here, if the global global the size of the market is unpredictable. It's maybe n number of $1 trillion. Yeah. So we can take that together and have the real world solutions. Translation research. It's like publication research is not appreciated, not regarded, either because of the funding agencies as well as the government, because they have to look after these problems. So that's our focus, and it's a great pleasure to have chance to listen you. And we will collaborate, maybe possible feasible in different areas, because we have great number of the instrumentation and the big teams and all sort of the infrastructure. It's like American standard. Simply I mentioned air conditioned laboratories and the genius science guys, students, from the master to the PhD, the postdoc to the assistant professors, work under the one group, and bulk of the, and I will give you the n number of opportunities to join the project as well. So that is also
Very
possible.
nice. Very nice. I will be very happy.
Yeah, sure. So that I welcome all of our participants as well. This is not the small opportunity, $50 million opportunity. Everybody can take part, and it's not limited to any country or any. So everybody can take part because of this government project. It's for industry welfare project. So everybody can just make sure that their contribution is appreciated there, and then that they are eligible to those kind of the participating in those kind of funds. Yeah, and projects.
Very nice.
Yes.
Thank you so much.
Yeah,
Yeah,
my
please.
question was my question was to Dr. Jury. What scientific methods did you all use to measure the concentration of the nanoparticles?
Actually, I'm just starting my PhD. And I would like to start to work with a metal remedy, metal substance. And I'd like to work with an institute in Romania for examining the sample which I use in my PhD. And I would like to use transmission electron microscopy.
Okay. Okay, thank you.
Most welcome.
Thanks a lot. Now we are moving for the next participant. Mr. Oliveira, are you available now?
Yes, I'm here.
Yeah. Please start your presentations and please note a time. You have a 10 minutes of time for the presentation.
Okay. Can you see my screen?
Yes, it's
Yes.
visible.
Yes.
Okay, perfect. Thank you. So, good morning, everybody. My name is Guilherme Santos de Oliveira. I'm PhD student from Dr. Terezinha. And today I will talk a little about my project in School of Pharmaceutical Science in the University of São Paulo. This project title is Influenza Vaccine, Technical, Scientific, and Regulatory Contribution to Patient Safety. So, talking a little about influenza virus, it can infect people and animals, and is divided in groups: influenza A virus, influenza B, C, and D. And the most important and common is A and B because it's most can infect people around the world. The subtypes are defined according to their surface structures and agglutinin in neuraminidase. And here I have influenza virus structure showing the hemagglutinin in neuraminidase. And for prevent infections with this virus, in Brazil, we produce the trivalent influenza vaccines that's composed of subtypes H1N1, B, and 3 and 2. So during vaccine production, and quality control is essential, especially the manager of this hemagglutinin, which is found on the surface of the virus. And right now, this measurement is done by single radial immunodiffusion, where the formation of the halos with addition of partners allows for the quantification of HA, this hemagglutinin. So the single radial immunodiffusion is effective but slow and expensive. And for this reason, using SDA's page and based on a methodology published by Harvey, I developed a faster and more affordable way to measure this hemagglutinin while maintaining the same level of quality. So this methodology stands out for its potential to deliver valid and efficient results with lower cost and time required for its implementation, as well as for its standardization. And here I have the methodology for Harvey. So the objective was develop a new analytical methodology for quantify this hemagglutinin content, validate this methodology according to Anvisa, the Brazilian Health Regulatory Agency, and conduct an antimicrobial efficiency test to verify the behavior of the influenza vaccine preservative in a second way of my project. So talking about the results in the development of the SDSPage analytical methodology for hemagglutinin quantification, I did acrylamide gel with 15 and 12% of acrylamide. What didn't work well, I couldn't see the separation of H1 and H2. And a second moment, I did with 10% using beta mercaptan and half to 96 degree. And I had these correct results with separates between H1 and H2 like the Harvey did. So I could consider this methodology development for quantify this hemagglutinin. Here I show the results that did not work. And after development, it's necessary to validate this methodology as recommended by the Anvisa. So I started to do some validations like the limits of quantification and limits of detections, where it's necessary to quantify the smallest amount of analytes in a sample or the concentration at which the expected visual effects can be observed. And here I have both situations. First, the limits of quantification. Here I can see H1 and H2 and can quantify correctly. And here I see just H1, what I can consider is the limits of detection. I analyze others parameters like precision and accuracy and specificity. So showing a little about each one. Here I have the results for precision. When I quantify correctly the 3 types of influenza virus and vaccine, and could quantify without any interference at this moment. So I could have sure the process is precision. For accuracy, I quantify in 3 different levels of hemagglutinin in the vaccine for the 3 types too. And here I can see the higher to the lower results in a mirror results too. And in both situations, I could quantify without any kind of problem. And for specificity, I did for the three types, subtypes, sorry, for influenza virus again. And here I can see the quantification with my standards, my vaccine, just influenza vaccine, and then another column with influenza vaccine plus HPV vaccine. And in both situations, I could quantify the same results, showing this methodology don't have interference from other kinds of virus in the moments of the quantification. After I finished this part of the validation and development of methodology, I looking for in another way for have the sure this vaccine is safety for everybody. So I did a stability test in influenza vaccine to see the preservative against different microorganisms. So the trivalent influenza vaccine undergoes stability tests in two types of study. Long-term stability study, where is the storage at correct temperature, and accelerate stability study, where these vaccines stay in the high temperature. So I did the inoculation of the sample with the microbial loads and the quantification of these microorganisms to monitor the stability of the preservative present in this product. In this case, the preservative is the thimerosal. And here showing the results of this test I have for the long-term stability after 12 months. I can see the number of CFU is declining. So this shows this preservative is working correctly. And in an accelerated stability study after 9 months, I see a little similar results, but with less effective. So it's not, I can see a degree, sorry, the loss of the efficiency of this conservant, but I can see they keep the levels of the CFU at the same level. So probably the temperature will influence in this efficiency of this conservants. So for the time I have is a little short, and I will publish these results. I will finish my presentation here, and I want to thank you for the opportunity to show my PhD results until now, and thank you again for everything.
Thanks a lot, Oliveira, for presenting your research work. Now, I would like to request Professor Mehul Patel for his comments or his queries. Professor Mehul Patel. Okay, I think Professor Mehul Patel is not here. So I would like to request Simon for her comment on his presentation.
Yes, it was a very good presentation. So what visualization agent did you use on the SDA plates that you showed us a while ago?
I'm sorry, could you repeat the question, please?
Oh, yes. A while ago, when you showed us the actual plates with the SDH page, right? What agent did you use to visualize the spread of the spots or the bands?
Okay. I used the chromacy for have color at this SDS page. And after this, I use a specific software from BioHard to visualize it and have these results in a specific software where I could correlate the size of the bands in my standard with my vaccine. So I correlated, and after this, I could do the correct identification of this hemagglutinin.
Right, yes, thank you.
Thank you.
Thanks a lot. Now next presenter is Mr. Mangesh.
Hello everyone. I'll just share my slides. I hope everybody can see my screen.
Yeah, it's visible.
Okay. So, hello everyone. I am Mangesh Kulkarni, a PhD student from University of Technology, Sydney, Australia. And before even I start, I want you all to do something unusual. Take a deep breath and hold it. Keep holding. Don't breathe. Feeling uncomfortable? A little anxious? Now imagine feeling like that even while you're not holding your breath. That's the reality for millions living with lung cancer and other chronic lung diseases. Lung cancer doesn't just take away breath, it takes away time. And my research is focused on giving that time back. So let's have a look on the statistics of lung cancer. And it is one of the deadliest cancers with around 2.5 million cases and 1.82 million deaths recorded just in 2022. And I'm pretty sure these numbers would be very high considering 2024 and 2025 statistics. And speaking about India, the 5-year survival rate for lung cancer is just 12%. And if we talk about the figures in Australia, the 5-year survival rate of lung cancer is just 17%. The problem is 85% of the diagnosed cases are advanced stage cancers, and this makes the treatment very difficult. So to understand this disease, the potential risk for lung cancer include majorly the tobacco exposure, which is smoking, and also the secondhand smoke. Both of these risks account for almost 90% of lung cancer cases. And nowadays, looking at the rising air pollution, it is one of the major risk factors as well. And some other risk factors include radiation exposure or occupational exposure and family history or genetic history as well. Speaking about the treatments, the currently available treatments are majorly, I've divided them into 3 different categories. One is the chemo or immunotherapy, which are mostly given together or individually, depending on the case and the scenario, and the radiation therapy as well as surgical procedures based on the stage of the cancer. But often these treatments are symptomatic and do not treat the underlying cause. This brings me to the heart of my presentation or this research, which is the use of phytoceuticals. One such phytoceutical is quercetin. It is a potent polyphenol flavonoid which has more than 50 distinctive biological activities, all the way from being a potential antioxidant, a potent antioxidant to an antiviral agent. But for now, we are focusing on 3 major activities of this flavonoid, which includes the antioxidant, anti-cancer, and the anti-inflammatory activity. So it is a yellow compound, just like curcumin, and it is abundantly found in variety of fruits and vegetables, and is majorly found in the fruits which are rich in vitamin C as well. But there is a limitation with the direct use of this uh, potent phytoceutical, which is it has poor solubility, poor stability, as well as low bioavailability. Speaking of numbers, the bioavailability of this drug given orally is just around 6 to 8%, which is not at all enough for the therapeutic efficacy. The answer for these problems would be in advance, giving it an advanced drug delivery, which could be a nanoemulsion. So for this project, we have shortlisted to encapsulate this drug as a nanoemulsion in a nanoemulsion. So we have multiple benefits of encapsulating this into a nanoemulsion, which includes the increased surface area because of its nano sizing, the improved solubility of quercetin itself, and enhanced cellular uptake, as well as targeted delivery and deposition, and controlled and sustained release. Because lung cancer is a progressive disease, and there is a very high need of controlled and sustained release of this drug. So speaking about the aim of this research, we aim to assess the in vitro anti-cancer efficacy of quercetin nanoemulsion and compare it against the activity of quercetin pure on the human adenocarcinoma cell line, which is shortly called as the A549 cell line. So this is the experimental flow we followed for our studies. So I would majorly divide this into two different kinds of assays, which would be functional assays and the mechanistic assays. In functional assays, we look for various hallmarks of lung cancer, such as cell viability, cell migration, and cell proliferation. So for cell viability, we do an MTT assay, which is required to figure out the maximum safe concentration of this drug, which is actually not killing the healthy cell line, but as well as on the same time is effective on the cancer cell line. So, and for cell migration, we can perform two different types of assays, which is scratch wound assay and a transfer chamber assay. In scratch wound assay, we grow the cells in a monolayer sheet on a 6-well plate, and then make a slight, tiny scratch using a sterile tip or a syringe, and then we look at the progress of this closure of the scratch, which will give us the migratory parameters of the cells when treated. Similarly, transfer chamber assay also gives us the invasive and as well as the migratory parameters of the cancer cells. And for cell proliferation, we can perform the colony formation assay. And once we have and the promising data from all these 4 or 5 different kinds of assays, we can actually look for the mechanistic, reasons for these effects, which include, so here we have applied protein expression and gene expression, where we have performed protein array using a human Excel oncology kit, which has approximately 96 proteins on the blots. It's very similar to Western blot. And then followed by, we also did a RT-qPCR for evaluating the gene expression. Coming to the results, first I would like to discuss the results from functional assays. Going to the dose which we selected through MTT assay was 25 micromolar. We have selected this dose because it was not killing, or it was not significantly reducing the viability of healthy cell line, which was the PCI NS 1.1 cell line. And I have not put the results here because of the limitation of slides and the time. So this is this graph shows the difference between the effectivity of quercetin pure and the quercetin nanoemulsion. And after finalizing the dose as 25 micromolar, we then performed scratch wound assay, colony formation assay, and transfer chamber assay, where we found that there is a significant difference between the migratory parameters or even the clonogenic parameters between the quercetin pure and quercetin nanoemulsion as well. After finding these effects and after finding these assays to be fruitful, we then were looking for the mechanistic explanations for these effects for reducing the hallmarks of lung cancer. And then we performed a protein array, which gave us multiple proteins. So I have shortlisted a few proteins for this presentation, but on the whole, I think we got 19 to 20 proteins, which were downregulated by this nanoemulsion very significantly. And some of them are included here, which includes a wide range of proteins from chemokines to interleukins, and even some proteins such as angiopoietin-1, which are responsible for angiogenesis of cancers. And then we performed a qPCR. So A549 cell line, the cell line which was used here is a KRAS mutated cell line. So it is very important to see if the treatment is actually downregulating the expression of KRAS and BRAF as well, because BRAF is the downstream signaling for KRAS pathway. And we could find that both quercetin pure and quercetin nanoemulsion were able to downregulate KRAS and BRAF. And interestingly, after doing a thorough literature search, we found that these 4 proteins, which are mentioned on the screen, are actually responsible for the poor prognosis of cancer, which means the 5-year survival rate highly depends on these 4 proteins expression. And to confirm these results, we further did a computational analysis through molecular docking and various other softwares. And we could see that there was a strong interrelation. There was a strong interplay between all the proteins and gene data which we got from our mechanistic assays, confirming that quercetin was actually downregulating these protein hallmarks. Talking about the future directions, we now think of expanding these studies on this study on various respiratory diseases, such as looking for the effect of pollution and how it actually is downregulating various parameters, which is elevating after there is an insult like pollution or inflammation or even remodeling, and looking for more mechanistic explanation like apoptosis assays and other inflammatory pathways, expanding the in silico studies. And once we have all good results from these studies, we would then definitely try to explore the in vivo efficacy of this quercetin nanoemulsion. This brings me to the end of my presentation. And first of all, I would like to acknowledge Mother Nature for giving us these beautiful phytoceuticals, my university, my supervisor, Dr. Kamal Dua, for all his support and motivation, and the association, Professor Dahiya, for his support and all the questions I asked him and his support, whatever he gave me throughout this journey to present. Thank you.
Thanks a lot, Mangesh, for sharing your research. I have a small query. Nowadays, a lot of research is going on secondary metabolite nanoformulations. So, is any product is available in market, same like what you are doing a research, like a secondary metabolite nanoformulations products are available in the market? Or what is the basic outcome of your research? Are you going to develop any product which you are going to launch in the market?
Yeah, so to answer your question, I would say quercetin is already being taken as a health supplement very widely and is available directly for purchasing from any pharmacies or even online, like on e-commerce website like Amazon. But I did not come across any nano formulation so far, because manufacturing a nano formulation and scaling up the batch size would be very difficult, which is a requirement to be studied. And speaking about taking forward the studies which we are doing at UTS, for now we have got very promising data from in vitro assays, and we would be taking this research forward and evaluating everything in the in vivo, and then definitely I would be interested, and I would like to take this forward to scaling up and actually having an industrial formulation for market.
Okay, thanks a lot. Thank you.
Thank you.
Okay, can I ask a quick question, Dr. Gupta there?
Yes, miss. Yes, Dr.
All right, great. Yeah. All right. So coming back to the quercetin, and you mentioned about the low bioavailability and the checking with the nanoparticles. Did you now compare that with, say, if you attach a sugar to the quercetin, that might make it more bioavailable? So any considerations on that?
Yeah, so actually, I haven't practically done an experiment using that, but definitely I've gone through some literature, which would, I think, because attaching some sugar or maybe any other salt form, converting it into a salt form, would definitely change its molecular form. And then the kind of effects we are expecting, then we might not get that. That's one of the reasons.
Yeah. All right. Thank you.
Thank you. Miss Cassandra's Ruiz Colon.
Hello,
Yeah, hi, Colon.
let me share the presentation. It is visible now?
No, it's not visible. Yes, now it's visible.
Okay. Well, hello, my name is Cassandra Ruiz Colon. I am a PhD candidate in pharmaceutical science at the University of Puerto Rico Medical Science Campus. And today I will be presenting about the development of robust dry nanosuspension via synergistic stabilization. First of all, before I start, I need to say that this presentation includes only non-confidential elements of the project. So specific experimental details and results are withheld due to ongoing disclosure and intellectual property review. As an introduction, there's a lot of active pharmaceutical ingredients that have solubility issues, and therefore low dissolution and low viability. There's several strategies to overcome these issues, but the nanosuspension formulation is one of these strategies that overcome this issue. However, nanosuspension is still face critical stability and formulation challenge would hinder their further development as novel pharmaceutical dosage form. In terms of my project, I use as a model a benzodiazepine-based bioactive BPB1, which is classified under the BCS2, which means that have very low solubility and high permeability. So if you try to mix my drug in pure water, it will never dissolve. And also, it has very extensive first pass effect, so have very pure viability. So there's a necessity to create a good strategy, in this case, to create a dry nanosuspension, in my case, using nanomilling and lyophilization. So the objective to make a nanosuspension is to reduce the particle size because the surface to volume ratio will increase. And according to Noah's wetness equation, the solubility and dissolution will also increase. And therefore, there will be an enhancement in permeability and viability. So in terms of the characteristic of the nanosuspensions or dry nanocrystal, they are made of pure drug nanocrystal dispersed in water. So there's no need to use carriers, and this reduces formulation complexity. But they are need to be stabilized, and at the end, you can have either crystalline or amorphous structure. So the general process to create the dry nanosuspension is to prepare the suspension with a stabilizer solution, add the milling beads, and the suspension, start the milling process, measure the temperature, and sieve to separate the nanosuspension from beads. Then you have to add the cryoprotectant, mix well, I usually sonicate, then freeze, place the sample in the lyophilizers, ground the solid sample, and do redispersion to make sure that my particles remain stable, same as original. I use dynamic light scattering to do the particle size analysis. So in order to do that, I evaluate several critical process parameters, such as bit size, milling time, motor speed, bit load, drug loading, type of stabilizer could be anionic, non-anionic, polymeric combination, and concentration of a stabilizer. And I use one factor at a time. So the objective was to achieve my critical quality attribute, the desired critical quality attribute, which was to obtain a particle size less than 150, polydispersity less than 0.2, zeta potential more than 30 MB, and redispersibility index less than 120. So during the evaluation of my dry nanosuspension, I did redispersion, solubility studies, dissolution, and solid state characterization. And if you see from this plot, I use a different bit size, and if you use a smaller bit size, they produce consistently smaller particle size. Finally, after several batches, I obtained one batch with that demonstrate the desired characteristic and robust colloidal stability. This nanosuspension was stable over 9 months. In terms of redispersibility, I obtained by dividing the particle size after redispersion and the particle size of the original nanoparticle. And for my nanosuspension, the redispersibility limits were below 120. I also studied the effect of media composition in different pH media, and both in pH 6.8 and there's something. Sorry, there's someone talking during my presentation.
Someone needs to mute.
Sorry for that. Okay. Specifically at pH 6.8 and pH 1.2, these buffers critically impact the stability of my dry and suspension. But one of the strategies is to use biorelevant media with surfactant and to develop a good solid dosage form with a protected matrix. As you can see from solubility and dissolution data, dry and suspension show enhancement in solubility and faster dissolution. In terms of the solid state characterization, you can see here that my dryness suspension, the morphology is porous, a sponge-like morphology, and characterized by interconnected pore, and it's very different from the pure drop. So in terms of the differential scanning calorimetry, you can see that the melting point of the pure draw disappear in my dry nanosuspension. In fact, the thermogram is very similar to the cryoprotectant. The same results was obtained by PXRD. The pattern of PXRD is very similar to the cryoprotectant. And also with FTIR occurred the same. And this is because I used a very low concentration of the drugs compared to the cryoprotectant. So maybe the cryoprotectant isn't masking the drug. However, in the Raman spectroscopy, there's a region in which none of the stabilizer or cryoprotector interfere. And you can see the orange is the drug, the green is the physical mixture, and the blue is my dry nanosuspension. And you can see that there's some peaks that disappear and the intensity diminish, that these could suggest a conversion to a morsel form. So as a conclusion, nanomilling with optimized stabilizer level produce synergistic stabilization of BPV1 nanosuspensions, and the choice and concentration of cryoprotectant prove critical for achieving a robust dry nanosuspension, enable progression toward pharmaceutical development. As a future work, I would like to do redispersibility, solubility, and dissolution in biorelevant media, and also to develop a dosage form and conduct preclinical studies. I would like to thank my major advisor, Sumita Tahia, and my thesis committee member. And also, I would like to thank the Puerto Rico Science, Technology, and Research Trust and the National Institute for Minority Health and Health Disparities for funding, and as a sharing facilities, the Molecular Science Research Center. Any question? And thank you for your attention.
Thanks a lot, Colin, for your wonderful presentation. I would like to request Dr. Simon for her comments on your presentation, please. Dr. Simon?
Yes, it was an interesting presentation. I like this slide when you showed us the actual suspension there. So I appreciated that. So thanks for the presentation.
Thank you. Now our next participant is Miss Andrew from AIMS University, Malaysia. Yeah, hi, Andrew.
Good evening, sir. Can you hear me? Am I audible?
Yes.
Could you see my slides, sir? Are my slides visible?
Yes, visible now.
Okay. 1 second. My screen is lagging. I'm so sorry. Okay. Very good evening, everyone. I'm Sarah Grace Andrew from Malaysia. And actually today I will be presenting my undergraduate research project, which is the solid dispersions of ketoconazole and approach to improve its aqueous solubility. So to start with, as we know, solubility problem is a very big problem, especially when we are developing new chemical entities. And we are constantly on the lookout for ways of and approaches of trying to improve their solubility. So we have various techniques and methods. For example, like now, the recent developments would be like in 3D printing and like nano suspension, like our previous presenter had already presented in her research project. So for my UG project, we decided to actually improve the solubility of ketoconazole, which is a BCS class 2 drug, via making it into solid dispersions. So we plan to actually use different carriers like PBA, PBP, and bird's nest powder. So as to the best of our knowledge, and I have done a literature survey, actually there's no one who has used bird's nest powder as a carrier for improving the solubility before. So I believe, as the best of knowledge, we are actually the first persons to actually do this work using bird's nest powder as a carrier. So we will be using these 3 different carriers in different drug to carrier ratios, which is 1:1, 1:2, and 1:5, and we'll be actually evaluating to see which carrier has the best solubility profile. And does bird's nest powder really have a potential in improving the solubility of our drug, and as well as future potential for other BCS class 2 drugs as well. So a thorough literature survey, which I have done, so far no one has ever used bird's nest powder in improving the solubility. They have used like PEG, and they have used PVP, and they have used Solid Plus. But so far, and all these studies are actually promising because there are improvements in terms of the mean dissolution time, in terms of the drug release when they're using these carriers. So actually this inspired us to use bird's nest powder, which is also naturally available, and we thought, why not? And it's also like, it's edible. I mean, people use it, and there's also various other benefits to bird's nest powder as well. So we decided to, and this actually inspired us to actually carry out this research. So as I mentioned before, we will be trying to, the ultimate aim would actually be obviously to improve solubility of our model drug, which is ketoconazole. But other than that, we also like to, identify which carrier has the best solubility enhancement and which carrier drug to carrier ratio it has the best results. And also we would like to evaluate the potential of bird's nest powder, whether it really does have potential in terms of improving the solubility. So in terms of methodology, so this is actually our plan of study. So it's divided into 4 stages. Pre-formulation, basically, we first tested the solubility of each of our carrier and our drug in different solvents. And we went for analytical characterization to determine the lambda max, phase solubility studies as well. And in formulation, we had prepared our granules. I would say our formulation is in granules and via solvent evaporation and kneading method. So I will be elaborating further in the later slides. And following after formulation, we had prepared capsules from those granules. So we put those granules in capsules, and we had gone for physicochemical characterization. Basically, we investigated their drug content and also performed the in vitro dissolution studies and kinetics analysis using software, DD Solver. And we also went for solid state characterization. We had only done FDR and UV. So phase solubility studies we had done as per protocol, and we had investigated at 2 different temperatures for 24 hours, which is 25 degrees Celsius and 37 degrees Celsius. And we had calculated the thermodynamic parameters, which was the Gibbs free energy, enthalpy change, and entropy change. So formulation, this is where it gets a little bit interesting. So only for PVP, we had done the solvent evaporation method, whereby we actually introduced our drug with PVP and dissolved it in dichloromethane after heating, and we have formed the granules after that. And for PVA and bird's nest powder, so the approach is a little different. So firstly, we had actually dissolved our model drug ketoconazole in DCM, which is dichloromethane, and then we introduced PVA or bird's nest powder, and we tried to rate it slowly until it formed granules. So it is a little different. So this is the kneading method. And following the formulation, after we had obtained our granules, we had produced capsules, and we went for the drug content analysis and in vitro dissolution study. So the lambda max was 222 nanometers, and I would like to actually take a note here. We had only done drug content analysis in triplicates. However, unfortunately, for dissolution study, we were not able to do it in triplicates due to time constraints because our UG project is actually just a few months. So that was one of the limitations which I hope to overcome in future. So as for the kinetics analysis and dissolution parameters, we actually computed using a software which is known as DD Solver. And moving on to the solid state characterization, we had performed UV interaction study and FDR analysis. So, and unfortunately, we were only able to perform 2 different types of characterization, once again due to time constraint and due to our lab not having adequate facilities to actually conduct the characterization. So this is actually one of the limitations. And moving on to the results and discussion. So actually, these are the thermodynamic parameters we had obtained earlier from our phase solubility studies, which is the Gibbs free energy and enthalpy change and entropy change. And as we can see, as I've highlighted here, all the values are negative. All the values are negative, and this indicates that actually all our carriers have solubilizing effect towards our model drug in the physical mixtures that we had actually formulated. So moving on to the drug content analysis, as we can see, PVP actually had the highest yield, especially when the drug to carrier ratios increased, as we can see here. The highest was 1:5 ratio with 150%. And the second highest actually bird's nest, which is actually quite promising, as we can see, is 80.36, 80.36 in a 1:5 ratio. But if we look at KPVA, when we had used PV as a carrier, as the drug to carrier ratio increased, the drug content analysis seemed to decrease. And moving on to the dissolution in vitro dissolution studies. So this is the data for KPVA. Unfortunately, even for in vitro dissolution studies, we were only able to do it for 1 hour due to time constraints once again. And so we actually found out that KPVA11 had a better dissolution profile as compared to our pure ketoconazole drug. But unfortunately, as the drug to carrier ratio increased for PVA, it did not show actually promising results. In fact, it was a little lower, much lower than our pure ketoconazole, in fact. And this is the data for PVP. PVP is very promising, as we can see. Actually, all the formulations in the different drug to carrier ratios, as the drug to carrier ratios increase, in fact, we can see a very good drug release pattern over 60 minutes. As we can see, actually KPVP15 is showing the highest drug release, which is the purple one at the top there. In just 1 hour, we are getting 28.46%. So in terms of PVP, actually KPVP15 was showing the most favorable profile. And moving on, this is actually the drug release data for bird's nest powder. And we are actually very pleased to say that bird's nest is showing quite an immense potential in improving the solubility and dissolution. In terms of, as we can see, the drug release is actually quite significantly improved in KBN11 as we compare it, and KBN12 as we compare it to the pure ketoconazole. Unfortunately, KBN15 is not able to have the good dissolution profile, but this actually indicates that if we tend to use a lower concentration of our bird's nest, it's able to actually achieve better dissolution, which is actually good in terms of economical standpoint as well for us. And this is the kinetics analysis, which I had done using the DD Solver software. And as per most of the formulations were actually exhibiting Cosmea-Papa's kinetics. And moving on, these are actually the dissolution parameters. And this is the UV interaction study in order to determine if there's any change in the peak or the lambda max. This is to actually detect if there's any interaction between the drug and the respective carriers. So as we can see in the PVA, all the peaks are actually aligned. So there's no interaction. And in terms of PVP, we can actually see there are some deviations from the peak and the lambda max. So the topmost peak is actually the one indicating a pure ketoconazole, and we can see there's actually some deviations. And this indicates there is some interaction between the drug and the carrier, which would actually be translating into a positive contribution to our solubility profile and dissolution profile. So this is considered a good interaction. As we can see, the highest, there is like a slope in the green peak, which is the PVP15, and PVP15 actually had the highest drug release in 60 minutes. And moving on, this is actually for bird's nest. You can see the peaks are all aligned at the same lambda max, which means that actually there's no interaction. And moving on to FTR analysis, this is for in regards to PDA. The topmost is for the pure ketoconazole. And as we can see, if the peaks, there is no interaction found. These are the peaks. I would like to draw your attention to KPVP. So as we can see here, there are actually some absence of peaks, especially we can see in the KPVP 15 formulation. KPVP formulation showing that actually there is a absence of peak. In tertiary, which actually, as per literature also, which I found out, it is actually beneficial in terms of dissolution profile. And moving on to KBN, actually the peaks are pretty similar. There are no much possible interaction except for the fact if we see very closely here for the KPVP15 spectra, we can see in between 3000 to 3500 there is a little bit of broadening of the peak, which is quite similar to the bird's nest, the green peak at the peak B. So which indicates it's actually a OH stretching. So that might be one of the reasons why KBN15 did not have a very favorable drug release profile in terms of its dissolution. So moving on to the conclusion. So actually we had successfully formulated the formulations using these three carriers, and it can be concluded that actually PVP had the best dissolution profile. Second best was actually bird's nest, and the least promising was actually PVA. And it can be concluded that bird's nest powder is actually very promising, and we can altogether use it for future research in different formulations, probably in different techniques as well. We can use it in 3D printing, we can use it in nanosuspension, try to understand how it works, try to see the dissolution profile of even other different drugs, BCS class of drugs as well. And also, due to the limitations of our setting, because it was an undergraduate project, we were not able to do it properly, like in terms of the time, in terms of the equipment available. And we did not even do a preclinical studies as well. So these are the improvements that can be made in the future. And if there is a chance and opportunity for that, I'll be really happy to actually continue with this. So these are my references. Thank you very much for your attention.
Thanks a lot, Ms. Andrew, for presenting your research.
Thank you, sir.
Now I would like to request Dr. Simon for her comments.
Okay, thank you, Dr. Gupta. Just one question
Yes,
to
sir.
Miss
Doctor.
Andrew. Yeah,
Yes,
when
doctor.
you when you said you mixed it with the dichloromethane, right?
Yes,
That boils at a relatively low temperature. So why did you add extra heat?
Just to ensure it's fully solubilized as well.
Ah, okay, all right. But did you check to see if the ketoconazole, the structure changed with the heat?
Actually, no. No, doctor.
Yeah, like if
Because
you had
we were
done-
in a very time constraint.
Yeah. Yeah, I was just wondering about the heat factor there when you heat it. Yeah, if that would have changed the structure and then changed the efficacy of the preparation. So that's something to consider then going forward.
Yes, doctor. Thank you very much for that suggestion. I will certainly take that in note when we are actually trying to do this work in the future later with better improvements as well. Thank you so much, doctor.
Yes. But the idea of using the birdnest soup, well, that is something I didn't even conceptualize. So that was really a novel idea.
Yes, doctor.
Thanks, Dr. Gupta.
Thank you. Thanks a lot, Dr. Simon. And thanks a lot, Andrew. Now our next participant, Miss
Thank you, sir.
Yulia Gabriela. Miss Yulia Gabriela. Is Julia Gabriela is here?
No, she's
Julia
not
Gabriela?
here. She's not here.
Miss Sarah, can you please remove your slides? Still, it's on sharing basis. Your slides up. Now we are going to the next, Miss Tehseen Khan.
Yes, I'm here. Just give me a moment. I'll share my screen. Thank you. Is my screen visible? Hello?
Yes, it is.
Okay. Yeah. So, let me just switch on my video as well. Sorry for the delay. Yeah. So I would like to first thank the APP organization for giving me the opportunity to present my research. So I'm Joita Tasin Khan. I'm a first year PhD student in Cancer Systems and Therapeutic Lab. I'm working under the supervision of Dr. Amit K. Tiwari, and I'm a first year PhD student of University of Arkansas for medical sciences. So I will start with the outline first. I'll first get started with introduction. So I will walk you through a brief introduction of the pancreatic adenocarcinoma, or also known as PDAC. So our rationale is to target PDAC with tubulin using quinoline scaffolds, followed by the key biological findings that we found with our proposed drug RA5, and finally our proposed mechanism of action with our so far results. So just to get started about PDAC, so PDAC is like an aggressive malignancy arising from exocrine pancreas. And as the PDAC develops, the normal ductal structures are usually dense and it's replaced by tumor cells surrounded by dense fibrotic stroma. And the very complicated microenvironment where the blood vessels are very compressed. It also limits the drug penetration, as well as it promotes immune evasion, which makes the drug's prognosis more poor. So as we look at the PDAC statistics, it occurs less in number, but the mortality rate is very high. And in 5-year survival, only 8.5% survives. Even if you look at the other GI cancers, we see that PDAC, we see the higher amount of death in pancreatic ductal adenocarcinoma cancer. And so we wanted to design some drug molecules based on some literature review where we saw targeting the quinoline scaffold with quinoline scaffold. When we target tubulin, we see micromolar IC50 death. It can inhibit telomerase, BCL2, as well as the FDA-approved drug like lenvatinib, topotecan, also zibotinib. They are like very strong tubulin inhibitors, as well as quinoline derivatives. So we we had 9 derivatives of quinoline 3 carbonitriles, among which we found RF5 to be very potent against certain cancers, particularly in pancreatic cancer. So we saw in pancreatic cancer, such as cancer cells such as PANC1 and MIAPACA2, we saw below 300 nanomolar concentration IC50. Where further we wanted to see like how it's actually more selective to pancreatic cancer or not. So we saw that compared to HEC293, which is a non-cancerous cell, it is very selectively cytotoxic, and compared to other 8 derivatives in this quinoline 3 carbonitrile series, RF5 is particularly very toxic. And we also tested it with 9 concentration, where we see like the survival rate is going down in dose-dependent manner. And then we also wanted to see its proliferation effect, where we see at 1 micromolar, the proliferation curve turns completely flat in both cancer cells. Then we wanted to also see its colony inhibiting effect, and we see with increasing concentration, RF5 is significantly reducing the colonies in dose-dependent manner in both of the pancreatic cancer cells, such as PANC1 and MIAPACA2. And we also looked at the morphological changes, where we saw that at 24-hour at 1 micromolar concentration, the drug is almost wiping out the cells. Also, cells are rounded up, which confirms the detachment. And also there is formation of blebbing, which kind of gives us a signal that the cells are going through apoptosis. We further looked at we tagged it with DAPI, OS, and then we saw that there is nuclear fragmentation also going through. So we were thinking whether it's going through DNA damage imaging effects. We also wanted to see its anti-metastatic properties. So we looked at its metastatic properties with wound healing assay, and there we observed that in both cancer cell lines, in over a 48-hour period, with increasing concentration of drug, the wound stayed open compared to the control in dose dependent as well as significant manner. Furthermore, we wanted to see its effect on the mitochondria, and we saw that the mitochondrial membrane potential is also going down in dose dependent manner, which is also observed in treatment with paclitaxel, which is a potent anti-cancer drug. So the mitochondrial membrane potential going down is like a key hallmark of apoptosis. Also, we see in PANC1 and MIAPACA cells there is nuclear condensation happening compared to the control. So we were also thinking that it's going through apoptosis, but we further wanted to confirm that. Then we'll observe the apoptosis in annexin PI assay with the help of flow cytometry, and we see that the apoptotic cells are significantly increasing in Q3 and Q2 region, which is early and late apoptosis, and there is also a little bit of movement in the necrotic region in both cancer cell line PANC1 and MIAPACA2, and this similar effects are also observed in paclitaxel, the positive control. Further, we looked at the mechanistic profiling, whether it's actually how is it whether it's going through intrinsic or extrinsic apoptosis. So we looked at some proteins. First, we saw BCL2, which is an anti-apoptotic protein, and we saw it's going down in dose-dependent manner. We also saw a little bit of increase in BAC, and which is a pro-apoptotic protein. And then we saw that caspase-7 is getting cleaved. The cleaved caspase is going up, and PAX we saw in one cell line, it's going up. Caspase-3 is going down. Also, we saw that PARP is getting cleaved. In both of the cell line, we saw a little bit of effect on that. We are trying to replicate, this study is still in an initial phase, so we are trying to replicate the Western blotting a little bit more. And then we also wanted to confirm with real-time PCR, where we saw that in both in Mia-PACA cells, the BCL2 is inhibited in a dose-dependent manner, and also we saw caspase-3 activation and also caspase-7 activation. Furthermore, we wanted to see if the target is actually tubulin or not, because we designed the compound to be a tubulin inhibitor. So we did docking, where we see a favorable binding in the colchicine binding site. Furthermore, we did tubulin polymerization assay with 3 micromolar of paclitaxel nogodazole, where we see RF5 is almost competing with nogodazole, which is a potent tubulin inhibitor. Then we did imaging with tubulin tracker, where we saw that beta tubulin is almost being inhibited with increasing concentration of RF5, and also we saw clear tubulin disruption compared to the control. Then we wanted to see, since it's inhibiting the tubulin, we wanted to see if there is any cell cycle arrest. So we did cell cycle assay with a flow cytometry, where we saw that cells are getting arrested in G2M phase, and we use nogodazole as a positive control here for our tubulin inhibition, and we see that cells are going through G2M arrest in dose-dependent manner in both of the cell lines. Furthermore, in the meantime, we also send the compound to NCI-60. So NCI-60 is a panel in US where they screen a drug against 60 cell lines, and we wanted to look at if the drug is selective against any of the cancers we are interested in, and we interestingly saw that it is very effective in HCD116, which is colon cancer, and we saw that RF5 is also very effective against colon cancer, HCD116. So also, I would like to mention that NCI-60 does not have the pancreatic panel, so we could not really compare with the pancreatic panel. But since we found its effects on HCD116, our lab was sending proteomic samples for on HCD116. So we also sent like RF5 for HCD116, and we saw multiple tubulin-related proteins are highly downregulated, which contributes to the microtubulin integrity as well as the maintenance of the cell structure. So we tested those particular genes in Mia-PACA cells in RT-PCR, and we saw that it's dose-dependently getting inhibited, and it's getting inhibited significantly, particularly the tubulin genes like TUBA1B, TUBA4B, TUB2A, TUB, RRM2, and CD44. RRM2 mainly contributes to the RNA biosynthesis, which further contributes to the tubulin polymerization, and CD44 is an immune marker which we found highly upregulated. So we were just interested, and we wanted to see its effect in RT-PCR. So till now, our conclusion is that RF5 is particularly targeting the tubulin-associated proteins, which is decreasing the alpha-beta tubulin monomers availability, and it's further it's further inhibiting the formation of the tubulin dimer and heterodimer formed from alpha and beta tubulin, followed by tubulin depolarization inhibition, as well as tubulin disruption, which is causing spindle defects, followed by G2M arrest, and which is causing activation of intrinsic apoptosis, extensive ROS formation, cleaved caspase-3 and 7 upregulation, as well as BCL2 downregulation, back upregulation. And this is contributing to apoptosis, and which is furthermore helping the cells clonogeneity, proliferation, and invasion to go down. But the study is still at a very initial phase, so we would like to see its mechanistic profiling more with more tubulin associated binding assays. So with that, I would like to acknowledge my lab, I work under Dr. Amit Tiwari, and these are our lab members and some of our summer interns. And I would also like to thank our committee member, Akin Burns Lab, Griffin Lab, also Dr. Rupak Pathak's lab. Thank you. With that, I would like to conclude my presentation.
Thanks a lot, Ms. Khan. Now, I would like to request Dr. Simon for her comments. Dr.
Yeah,
Simon?
yes, thanks, Dr. Gupta. Yeah, it's current research because the pancreatic cancer has been really making inroads, and it doesn't seem as though there's much that can be done once it's diagnosed. And then also the way of its detection, sometimes when people find out, it's already at stage 4. So it's really current research. So thanks for the presentation there.
Thank you so much.
Can I ask one question?
Yes, sure.
Okay. Nice presentation, and I appreciate your understanding about the molecular biology of cancer. And I think the cancer concern will be growing because of the numerous reasons, like residues in the food, like agrochemicals, then packaged food together, like some kind of colors, flavors, and some chemicals too. Yeah, some agent, and like for example, another concern is like overdose of the medicines together. So in lifestyle stress and stress after the age of in the age of the AI will be not uh, reduced, but it will create more stresses in many billions of people life because of the layoff of layoff is now very evident and is expected to reach around 60-70%, maybe within the just 1 or 2 or 3 years. And because of the automation in industries, workers protest together, they will also lose job, and scrolling together, stress and family drama together, that will be also reasons for a number of different cancers. And the cancer doesn't come alone. It comes like consortia with other metabolic disorders together, and sometimes nerve damage and this. And maybe I personally believe that in the near future there will be new diseases will be also appear. It will be like a lot of serious efforts to diagnose them. Okay, and I would like to discuss one example. It is also detected a cancer biomarkers, but they have the both hands like unbearable pain. They can also not bear the fan air as well, and also they together they have diagnosed with cancer biomarkers. So this it took 2 months. It's one of the colleague story is because of that I also discussed that story. Like so for example, neuron there's some like coating on the neuron, some kind of, and then cancer biomarkers, and then pains on the both hands. It's like maybe one hand pain may be very common, maybe it can be kind of another thing, but both pain give the some kind of the insight about the brain things. They again revise different kind of the diagnosis.
Yes.
After 2 months, but there is no name for the disease because that doctor has not studied that kind of disease or never encountered with such kind of patient. So that is that's why because I know that person from the long before, like 20 years, more than 20 years. So I know that what is the mental setup of that man. For example, negativity together with some kind of stress or anxiety or unpredictability, uncertainty together, some regrets together, and bad thinking about others, like not bad wisher, I would say. So and how the political or some kind of cunning or some many psychological issues together. And I believe that as per my knowledge, when I came to know him and before as well, he must be similar from childhood or some. It's like long then 20 years or 30 years of together, it caused this. It's not like metabolic disorder doesn't pop up in the 1 month or 1 year. It takes long history together. And I will not talk about any name or personal history about that, but he been isolated guy. He not been among the peer colleagues, and just like one maybe only family guy. So not non-social, maybe it's just pretend to be social. Sometime maybe gone through the serious setbacks, so that kind of things together. So it can not only alone about the chemicals, I would say. Cancer is not alone about chemicals. It has numerical reasons that together manifest cancers, like acidic condition together, some cases. And so that's why I think I would recommend you to look after uh, the serious concern about the cancer. So to make aware, because new age people are going to be more prone to they be like fast food, junk food, and like pizza burger trend is coming together, and stress will be like as I described, the billion people will be stressful, and then scrolling together, like imbalancing the hormones, like dopamine is there as well as cortisol is there. So it's upsurge. Both upsurge is not, it's like contrast, right? If there is the dopamine, there should be no place for the cortisol. There is scrolling, they can see the some like pleasures things, and they get the some like small boost of dopamine, and but they are stressful hormones that's upsurge. That chemistry is going to, because social media is also will be equally contributor to even it at age groups or economic class will be not the, but it it is of great concern. I would say please keep focus and stay focused on the career, because sometimes researcher I part know they are like discontinued, or like sometimes they disperse their focus, and that's make them like nowhere. So keep focus and go deeper molecular biology level studies, because uh, biology is like a new light for the people who don't know the biology. This is the universe, new universe. When we dig deeper, that's we can dive into the deep. But the common people don't know the biology. B, they don't know the B. They don't know cell definition. They don't know the even the, and they still living the biology body, and but they don't know the about the even B about the biology. So that's the reality of the world. Most people means even the professional people biology. So our job should be make the greater awareness to understanding the biology, at least basic, to protect their psychology as well as their body chemistry. So that should be also go the parallel, not only the focus on the research publication. Publication will be okay, pleasures, okay, happy for the one day. But what about the okay, maybe 1 week happiness? But to
Yeah.
keep the greater happiness and the like from the voluntary services, some kind of the podcast and interviews and some speeches to the large gathering, to their understanding way, simplifying way. Like Albert Einstein said, if you can't explain with simplified formulas, you don't understand. So when we understand, we can simplify this, like break down and make them ensure that even illiterate people can understand the
Yes.
concepts. So you can fly with both wings. So it will be great for your career, professional career, and be as a global leader with this organization, as well as together grow, support each other, yeah, grow each other, yeah, together. Thank you.
Thank you so much. Yeah, absolutely. I totally agree. And uh, just what you said, that it's very hard to treat. And as we know, it just looks like normal cells. It evades like our immune cells. So it's very interesting to study cancer, because we are always facing drug resistance with the already available drugs. So there is always research opportunities for us, and with this we can also help contribute to healthcare. And I deeply acknowledge your words, and thank you so much for the kind words.
Yeah, that's true. Like the multi-drug resistance is not alone for the pathogens, but it is also for cancer. Because of that, it's also biological cells. They can have that kind of mechanisms. It's very interesting. And we also work on the multi-drug resistance anti-cancer, especially antibacterial agents, to address like issue about multi-drug resistance. At even new n number of the antibiotics comes or chemicals comes, the antibacterial, the bacteria can just deal with that enzymes simply by enzymes. So that like antibiotics is not permanent solution and not going to give the great hope. So we are combining like some kind of the antibacterial agent with the together nano composites to do that, like the permanent solution like that. So in the future we can collaborate about the deeper understanding of science, because we work for science, not for the publications, because we are already reached to the thinking that the publication, and so far the whatever publication we did for the, like, who can add the science, but add the deeper science. Now the time is for deeper science. Yeah.
Yes, definitely.
Thank you.
Yes. Thank you so much for your kind words. I really appreciate it. Okay. I'll just stop sharing.
It's critical role which the diagnostic labs can play. But the challenge which I found was that the diagnostic business in India is very different from diagnostic business worldwide, because the other countries are far more regulated. So the diagnostic play is very different. In India, it's very price sensitive. So obviously, the services that you design and
I'm sorry, was that question for me?
પણ એ કેટલા 150 જેવા હતા તો આપણે થોડાક એનાથી ઓછા વાળું લઈએ તો ગઈ વખતે બુકલેટ તૈયાર કરી ત્યારે એની અંદર 2-3 પ્રતિ
É a mesma coisa. Eu fiquei na dúvida se
రేపు మార్నింగ్ చూద్దాం రేపు వస్తుందా
కుదిరితే వస్తా మేడం
ఏం గెట్ టుగెదర్ అమ్మ ఫ్రెండ్స్
మా ఫ్యామిలీనే మేడం యాక్చువల్లీ ఇప్పుడు వెళ్ళాలి
ତ ସେ କଥା ମୁଁ ମୋ ସାର୍ଙ୍କୁ ଜଣେଇନି ଯେ।
Colleagues, we have just binded out our last oration session. So let me start the valedictory now.
गुड आफ्टरनून
Good
दहिया
afternoon, sir.
सर
How are you, sir?
बस भाई
Good morning, Dr. Daya.
Good morning, sir. How are you?
Good, thank you.
Sir, good afternoon, sir.
Good afternoon, sir.
गुड आफ्टरनून सर
Good afternoon.
Good afternoon, sir.
Good afternoon.
Good afternoon.
My slide is visible to all?
No, not yet.
No, sir, no.
Yes, we're seeing it now.
Yeah, sir.
And the movement of content is there? Is the content is moving?
Yes, yes.
Yes.
If anything is not moving, just shout in between so that we can go. So okay, I welcome all of you in the virtual valedictory ceremony of the first day of APP 14th Annual International Convention, which is our third Indo-Korean conference. Tomorrow will be the physical face-to-face day of the convention. So today we will conclude it with the virtual award ceremony. So I welcome all of you again in this session. And today we are commemorating the World AIDS Day supporting WHO initiative. This conference is hosted by Vijaya Institute of Pharmaceutical Sciences for Women. So before announcement of awards, we have certain other announcements. We have new international branch and EC members. We have merged our APP Ethiopian international branch and broadened it to the APP African international branch now. The president, secretary of the APP Ethiopian international branch is promoted as president, Dr. Raghvendra Yerla Kada, who belongs to Jimma University, Ethiopia. Our new secretary has joined Dr. Ibtisam Hanachi from University of Monastir, Tunisia. New EC member has joined the APP American International Branch, Dr. Satish Kumar Vimula, who belongs to University of Mississippi, United States. Dr. Hanachi, you are here with us? No. Dr. Vimula?
Yeah, Dr. Balaji is.
Yeah, I'm available.
Dr. Vimula is not available? Oh, you
Dr.
can
Dahi,
share.
I'm available.
Yes, I am available.
Yeah. Okay. Thank you. New vice presidents and joint secretary, APP Punjab State. New vice president is Dr. Suresh Kumar Beniwal from Sri Sukhmani Institute of Pharmacy, Derabasi. Dr. Suresh, you are here?
जी सर आई एम हियर जी जी
Okay. APP Uttar Pradesh State Branch. Professor S. Sadish Kumar from ITS College of Pharmacy, Muradnagar, has joined as the new Joint Secretary. Professor Archana Mehrotra from SRK University, Bhopal, has joined as new Joint Secretary of APP Madhya Pradesh State Branch. And Professor Vittal Chopde, PES Modern College of Pharmacy, Nigadi, Pune, has joined as new Joint Secretary of the APP Maharashtra State Branch. We will start from our highest award, which is for lifetime professional contribution. And this award goes to Professor A.N. Nagappa, who is retired from the services and presently on visiting professor post at School of Pharmaceutical Sciences at Behra University, Solan. He is ex-director of Amity Institute of Pharmacy, Amity University, Gwalior. Before going further, if Professor Kiranmai is there? Professor Kiranmai?
Yes, sir.
Yeah, you can brief about Professor Nagappa, please.
I received Sandeep sir's one, sir.
So, earlier one is not with you?
No, sir. Actually, one you shared, no?
So, I sent the CV. Okay, anyhow.
Yes, sir. One CV. 1 minute,
You
sir.
have?
1 minute. Yes, sir.
Yeah. You can.
Yes, sir. Sure. Good afternoon, all. This is Dr. Kiran Mai, St. Paul, Principal, St. Paul's College of Pharmacy. Introduce Ananta Naik Nagappa, sir, on this occasion. And first of all, congratulations, sir, for the Lifetime Achievement Award 2025 from this prestigious APP 14th Annual International Convention. So Professor Nagappa, sir, is a professor, visiting professor, School of Pharmacy, Behra University, Solan, and Himachal Pradesh, India. And Sir's mission statement is to learn, practice, and teach highest quality of pharma-related knowledge and education. He is the founder president of Association of Community Pharmacists of India. And Sir pursued his Bachelor of Degree from Government College of Pharmacy, Bangalore, PG from Government College of Pharmacy, Bangalore, PhD from Pharmacology Discipline from BITS Pilani, Rajasthan, and DSc Honors from International Agency for Standards and Ratings. And various academic positions he held at different era, having vast experience, academia, and also industrial experience. In industry, he positioned as research coordinator in Regional Research Laboratory, Jammu, and quality control inspector in Kanfa Labs, Bangalore. And coming to projects, so he handled various funding projects like membrane phenomenon in drug action, pharmacological screening related, and chemotropism and pharmacological screening of Cassia tora. So various projects, funding projects, sir has handled successfully. And under Sir's esteemed guideship, 6 PhD candidates got awarded, and 3 people are under progress. And he guided, supervised 110 post-graduation projects and 80 B Pharmacy students also he guided. And academic honors and awards of Nagappa, sir. Lifetime Achievement Award, International Scientist Award in 2020 in Engineering, Science and Medicine, dated on 19th and 20th December of 2020. And sir has given a lot of invited talks at various platforms of international and national, like pharmacy practice Yeah, in 2012 in USA. Likewise, sir has visited various universities in abroad, right from Singapore, Malaysia, USA, Switzerland, Netherlands, around 19 different various universities sir has visited. And on his credit, textbooks are there. Surface activity in drug action and emerging trends in pharmacy practice are few of them. And even sir has expertised in clinical trials also. And various publications on his credit. So almost 50 plus publications are there. Till now, 62 publications are there on his credit, and out of which various national publications and international publications are there in indexed journals. And sir has presented many important topics in various platforms, conference presentations on his credit, and almost more than 10 membership professional society memberships are there. And likewise, if we list out, a lot of accolades are there on his name, and is a very renowned notable person in pharma field of education. So once again, I'm feeling privileged and thankful to Rajiv Dahiya sir for giving this opportunity with this note, such a wonderful person to on this platform. Thank you, sir.
Thank
Okay,
you.
Professor Nagappa, you are with us, so you can
Yeah,
share
yes.
your views.
Yes. See, the awards and rewards only make us to visit and motivate us to do further work. So I'm thankful to Dr. Rajiv. In fact, last 3-4 months, I remained at home and was not doing any job. And when Rajiv Taya called me and he wanted to honor me by award, it reminded me of my responsibility. Again, now I would like to start my working in a better manner. So work should not be stopped. And I'm thankful to APP and to Dr. Rajiv Zaya for giving me and reminding me my responsibility. Thank you, Dr. Rajiv Zaya. Thank you very much.
Congratulations, sir.
Thank you.
So we will move next. So first we will cover the international category awards. I would like to request those who read the CV just to conclude it in 30 to 45 seconds only, please. So Outstanding Achievement Awardee of 2025 year is Mr. Vishal Pathak from USA. Mr. Vishal, you are with us?
Yes, Dr. Daya.
Yeah. Before coming to Mr. Pathak, I would like to invite Professor Rita Moria, who is Vice President of APP MP State Branch, to brief about Mr. Pathak.
Hello, sir. Good afternoon. Am
Good
I
afternoon.
audible?
Yeah.
Okay. Thank you, Daya sir, for giving me privilege to brief about Dr. Vishal Pathak, sir. Mr. Vishal Pathak is a nationally respected leader in healthcare technology with more than 18 years of experience improving the systems that manage how Americans receive care and how their medical bills are processed. His work strengthened the digital backbone of major health organizations, making the flow of information, payments, and care coordination faster, safer, and more efficient. Supported by top-tier credentials including PMP, PMI ACP, FAHM, and multiple Agile certifications. He has led major modernization efforts as CVS Health, Blue Cross, Blue Shield companies, Anthem United Health Group, Optum, and others. Mr. Pathak is also an exceptional mentor who has trained groups of pharmaceutical and healthcare professionals in agile practices. Smarter claims, processing, and modern payment approaches. His teaching helps team reduces error, prevent delays, and control avoidable healthcare costs. Benefits that ultimately help patients save money and avoid high insurance expenses. His leadership is anchored in a strong human purpose, improving the everyday experience of people who depend on the healthcare system. Beyond industry work, he has served as visiting faculty, consultant, and advisor across the universities and global programs, guiding the next generation of healthcare innovators. His career reflects strategic thinking, technical expertise, and a genuine commitment to making healthcare simpler, more affordable, and more human. So I congratulate Vishal Pathak sir for this achievement. Thank you, sir.
Yes, Mr. Pathak, if you want to share, you can share your views.
Sure. Thank you so much. Good afternoon, everyone. Dear President, honorable members of APP. It's a real pleasure to be here. I'm deeply honored and extremely grateful to the association for presenting me with this esteemed award. For a quick introduction, my name is Vishal Pathak and I am a technologist. For the past 2 decades, my professional journey has been driven by a singular purpose: to improve healthcare outcomes for every individual through cutting-edge technologies, analytics, and agility. Addressing ongoing challenges with healthcare affordability and accessibility is essential, and I feel truly privileged to dedicate my work to this noble cause. My sincere gratitude goes to the President and Professor Tiwari for providing the opportunities to collaborate, and I look forward to serving this association as an active member in every possible way. Thank you.
Thank you, Mr. Pathak, and congratulations for the award.
Thank you so much.
So our next award is Distinguished Researcher Award 2025, and this award goes to Professor Somnath Singh. I again request Professor Moria to brief about Professor Singh.
Thank you, sir, for giving me this opportunity. Dr. Somnath Singh is a highly accomplished pharmaceutical scientist, educator, and researcher with an exceptional record of excellence in pharmaceutics and drug delivery. He has led groundbreaking projects in smart polymers, sustained release systems, and novel ocular therapeutics, securing major grants including funding from the National Institute of Health, the Health Science Strategic Investment Fund, and several prestigious faculty development programs. His notable research achievements include the development of innovative hydrogen sulfide-based microparticle delivery systems, exploration of novel glaucoma therapeutics, and pioneering work on polymer-protein interactions and skin absorption models. He has successfully completed multiple competitive grants totaling several $100,000. A distinguished educator, Dr. Singh has earned 6 Educator of the Year award between 2009 and 2025. An extraordinary recognition of his dedication to teaching, mentorship, and academic leadership. He is also a respected author, having co-authored the widely used textbook Pharmaceutics, Basic Principles and Application to Pharmacy Practice, published by Elsewhere, first and second edition, along with influential book, chapter in drug delivery, dermal absorption, and medicinal chemistry. With over 3 decades of his excellence in research, teaching, and scholarship, Dr. Somnath Singh continues to shape the future of pharmaceutical sciences through innovation, leadership, and a lifelong commitment to advancing healthcare. So I congratulate Dr. Somnath Singh, sir. Many congratulations, sir. Thank you.
Professor Singh, you can share your views.
Thank you so much for the kind introduction. Yeah, I am grateful and I feel honored. I humbly respect this award. And I am thankful for award committee for their time involved in selecting me as a award recipient. Otherwise, life is a journey, and I'm more than 30 years. I started my journey 7 years teaching in India, and then in the USA, about 22 years. So at this time in my life, this award is a great milepost. Thank you so much.
Congratulations, Professor Singh.
Thank you.
we'll move to the next award, which is Innovative Researcher Award 2025. And this award goes to Dr. Sandeep Lohan, who is scientist at Bristol Myers Squibb, New Jersey, USA. Dr. Lohan, you are with us? Dr. Lohan? Okay, see, he may not be available. So, but I will still request Professor Kiranmai to read
Yes,
few
sir.
lines about him. Professor
Yes.
Kiranmai?
Yes, sir. Thank you,
You
sir.
can brief Dr. Lohan.
Yes, sir. So congratulations, Dr. Sandeep Lohan, sir, for getting this award. So Lohan, sir, is from USA. So sir's education accolades are like right from B Pharmacy from Kurukshetra, Haryana, Masters from Manipal University, Karnataka, and he pursued his PhD Pharmaceutical Sciences from JP University, Information Technology, Solar, India. And coming to his research credits, so he has vast experience as scientist, professional experience as scientist lead discovery and optimization from Bristol Myers, Princeton, New Jersey, USA, and postdoctoral research scientist from Massey Cancer Institution, Virginia Commonwealth University, Virginia, USA. And he is expertise at development of macrocyclic peptide therapeutics for neurofibromatosis type 1 disease using mRNA display, and a postdoctoral research associate from Rinker Health Sciences Campus, Chapman University, USA. So the topic is development of orally active peptide-based anti-infective agents and development of peptide-based trans-infection agents. So scientist two and other doctoral research from JP University on development of structurally small antibacterial lipopeptides and peptidomimetics. And sir has notable leadership skills and communication experience. Like he led a team of 6 people to successfully manage a CRO project. So 9 years of independent and team-based project management experience. And sir has vast technical skills. A few of them are like well versed with the handling of various hyphenated techniques, including MALDI TOF, and also technically very sound person with updated knowledge. And 30 publications of national and international on his credit, and 4 patents are on Sir's credit, and Sir presented 6 on international and national platforms of his conference papers. So with this brief introduction, once again, I congratulate Dr. Sandeep, sir, for this award. Thank you, sir.
Congratulations. Yeah, congratulations. And I'm thankful to Professor Rita Moria, Vice President APP MP State Branch, and Professor Kiranmai Mandwa, President APP Telangana State Branch, for briefing the profiles. Thank you. So we will move towards our next award. This is Young Achiever Award 2025. And this award goes to Professor Ying Hua Wang, who is the research professor, Institute of Oriental Medicine, College of Korean Medicine, Dongguk University, Seoul, Republic of Korea. He accepted this award in the morning session when he delivered the lecture today via virtual mode. So we congratulate Professor Wang for the achievement. So next award is Young Talent Award 2025, and this award goes to Dr. Camelia Genciana Kubuk Judge. I'm sorry if I'm not able to pronounce the name correctly. She is a homeopathic physician as well as PhD research scholar at Department of Pharmaceutical Sciences, Faculty of Pharmacy, Western University, Vasil Goldis, Arad, Romania. Dr. Camelia, you can share a few words with us about your journey.
Hello, I want to thank you very much for this award and for the trust you have placed in my work. My scientific journey began with a deep desire to understand the human being in its fully complexity, which led me to complete my studies in both medicine and pharmacy. These two perspectives shaped the way I look at healing, both scientifically and holistically. Later, my path expanded to the field of homeopathy, a discipline that opened a new horizon of questions. My search for deeper understanding took me to India, where I had the privilege to learn from Dr. Rajan Sankaran, one of the most renowned homeopaths in the world. His extraordinary clinic results awakened in me a transformative question. I wanted to understand how homeopathic remedies interact with organs, tissues, and cells. What active principle might truly be present? Nanostructures, informational patterns, or subtle signals capable of influencing biological systems? And today, this question continues to guide every step of my research. The question is no longer whether something is present in homeopathic remedies, but what the something is and how it works. Because once we discover that, we don't just understand homeopathy better. We understand life, healing, and human potential at a deeper level. Thank you.
Thank you, Dr. George. We will keep in touch for the future conferences.
Thank you, for sure.
Congratulations. So now the international awardees with in the student category. So I'm seeing some green line on my screen. Are you also seeing the same?
Yes, sir. Yeah.
Yes.
I don't know from where it came.
Yes.
Okay. Anyhow, so first, we have selected 5 to 6 unique talent awardees from international community. First awardee is Mr. Kyung Jae Lee, who is the undergraduate student of College of Korean Medicine, Dongguk University, Goyang, Republic of Korea. And he is recognized for his excellent academic performance and research skills, supported by his supervisor. Mr. Lee, if you are with us, you can share your views.
Hello, everyone. Good
Yes.
afternoon. My name
Yeah,
is
yeah.
Kyungjae Lee, and I'm currently a third-year Korean medicine student at Dongguk University in Korea. And I am now conducting research at the Helmholtz Center for Infection Research in Germany for studying microbial metabolites. And it is a great honor to receive this reward from the Association of Pharmacy Professionals. I would like to express my sincere gratitude to my mentor, Jing Wang. And this award will motivate me to further advance my research on the interaction between microorganisms and their metabolites. And I want to contribute to this achievement to field of therapeutic development for the future studies. Thank you very much.
Congratulations, Mr. Lee. So before moving forward, I'm just stop sharing and again start sharing so that this green line will not appear. Thank you. Are you able to see the screen?
Yes.
Okay. So we'll move next. Our next candidate in the unique talent category is Miss Sarah Grace Andrew, who is the PhD student of Faculty of Pharmacy at AIMST University, Kedah, Malaysia. So, congratulations, Miss Andrew. If you want to share your views, you are welcome.
Good evening, sir. So thank you so much for this opportunity. I'm incredibly grateful. I'm Sarah Grace Andrew, PhD student at Faculty of Pharmacy, Ames University. Actually, I've just completed my B.Pharm and now I'm doing my PhD under the guidance of Dr. Floria, and I'm incredibly grateful for his support and guide throughout this journey. And I think without his help, I actually would not be able to actually get this award. So thank you so much, sir. And thank you so much for nominating me for this award. Thank you.
Okay, congratulations, Miss Andrew. Professor Floria, you want to say a few lines to for the student?
Yes, sir. Thank you so much for giving me this opportunity to speak about her name is Sarah Grace Andrew. Actually, really, she's gracious, I'll say. I could see her for last, it's about 5 years now. From foundation, then she took admission in B.Pharm, and throughout her B.Pharm, she received almost a four, the CGPA 4 out of 4 itself. So it's 3.99, so exactly same. And the biggest achievement of her, that's a commendable, I'll tell you. She's the only student I had so far in my life, I can say, I think, who has done more than 60 massive open online courses, the registered one, that too from a very top-notch universities and all. So that's a really commendable job, and she's very hardworking, very sincere. Like, in fact, now her PhD has she has already enrolled, and just in the mid of the the journey, she's in PhD, and she is she's a very workaholic, I'll say. So she's the pride of our university, I would say here. So I congratulate here at this platform, and I'm thankful to the APP for considering her candidature and looking on her credential and achievement, and to offer her this Unique Talent Award 2025, which she really deserves. So thank you very much, sir. Thank you so much. And thanks to our student, Miss Sarah Grace Andrew also. So, putting our collar high. Thank you so much, Miss Grace. Thank you.
Thank
Thank
you
you
so
so
much,
much.
sir.
Yeah.
Okay, congratulations, Miss Andrew, as well as Professor Floria, for
Thank you,
guiding
sir.
such a brilliant student.
Thank you, sir.
I want to tell about Professor Floria is leading as president of APP Malaysian International Branch, and he has been continuously supporting in nominating students from Malaysia. And so, thank you, Professor Floria.
Thank you, Professor Rajiv. Thank you so much.
So we'll move to our next award of unique talent category. And this award goes to Mr. Gulharme Santos de Oliveira. He is a PhD scholar at Faculty of Pharmaceutical Sciences, University of Sao Paulo, Brazil. Mr. Gulharme, if you are with us, you can share your views. I think he's not there.
Sorry, Guilherme is
Yeah.
not
So
just here.
his supervisor is there, Professor Terazina.
Yeah. So
Yeah.
thank you very much. In his name, I am certain that he will be very, very happy with this situation, with this great situation. Thank you so much, Professor Rajiv. Guilherme is a very hard student, and also he is not able to be here, but I will try if he can give us a message now.
Okay, it's okay. At last we can wait when we will remain for 10-15 minutes. So he can be at that time. He can share his views, no problem.
Thank you very much.
I have one request, Raju ji.
Yeah.
So
So
please
everybody
should
should
start
start
the
the
camera
camera
because it's like some
getting recorded. So please request him.
Okay, if everybody can start their cameras, it will be nice. It is a recorded session. Okay, I'll move next.
Okay, thank you. I sent
Yes,
him
congratulations,
message,
Professor
but not
Teresina.
possible. Thank you.
Next Unique Talent Award goes to Mr. Mangesh Pardeep Kulkarni, who is a PhD scholar at Department of Pharmacy, Graduate School of Health, University of Technology, Sydney, Australia, for his excellent academic achievements and research output. Mr. Mangesh, if you are with us, you can share your views.
Sure, Professor Dahiya. Thank you. Thank you very much. Thank you for this honor. I'm truly grateful for this award. This platform is remarkable, and I sincerely appreciate the rationale behind supporting emerging research talent. I would like to also sincerely thank my mentor, Dr. Kamal Dua, the association for nominating me, and particularly you, Professor Dahiya, for all the support and consideration. This recognition inspires me to keep pushing the boundaries. Thank you. Thank you very much.
Thank you, Mr. Mangesh. Congratulations.
Thank you.
So next Unique Talent Awardee is from Puerto Rico. She is Miss Cassandra Ruiz Colon. She is a PhD scholar at Department of Pharmaceutical Sciences, School of Pharmacy, University of Puerto Rico. Miss Colon, if you are with us, you can share your views.
Hello, I am very grateful to receive this award. As my trajectory, I studied chemistry and a master's degree in pharmaceutical science. And then I worked several years as a forensic toxicologist, which is very different from pharmaceutical. But then I discovered that I love teaching, research, and I wanted to use my skill to help people and contribute to the development of the new medicines. So I decided to start my PhD. So now I am a PhD candidate, a mother of 3 kids, a wife, and a full-time student. So I am very grateful for that. Thank you.
Okay, congratulations, Cassandra, for this achievement. So we'll move next. Okay, this completes our international category. We will move to the national category. First, faculty level awards. So first awardee is Dr. Archana Mehrotra, who is principal.
Can I have the break when my turn is there?
Yes.
I need short break. When will be my turn? I just have a quick convey some regards to some colleague. I will return back. So
okay, no problem. No problem.
Yeah.
So we have Professor Archana Mehrotra, who is principal at Sarvepalli Radhakrishnan University, Bhopal, and she is honored with Distinguished Teacher Award. And this award will be given tomorrow in face-to-face mode, as she is one of the speaker of the second day of annual convention. So congratulations, Professor Mehrotra. Next award is Principal of the Year Award 2025, and this award goes to Professor Marnal Sirsat, who is Principal of Dr. Uttam Rao Mahajan College of Pharmacy, Chalisgaon, Maharashtra, for his wonderful pharmaceutical administration potential. So I invite Professor Sirsat to share his views.
Hello, sir. Thank you. Thank you for the Night 14 Indo Conference hosted by Vijay Institute of Pharmaceutical Science, Vijayawada, and also thanks to the Rajiv Dahiya sir and APP. Thank you for giving the award, and thank you for appreciation for APP. Thanks, sir.
Okay, congratulations, Professor Sirsat. Our next award is Young Investigator Award 2025, and it is no other than Professor Balaji Pandyan, who is a Professor of Pharmacology at School of Pharmaceutical Sciences, Wells University, Chennai, Tamil Nadu. Professor Pandyan, you can share your views.
Sir, Respected dignitaries, esteemed members of the Association of Pharmacy Professionals, especially I want to thank to Raju Dahiya sir. I'm truly honored to receive the Young Investigator Award. This recognition means a great deal to me. And also I express my sincere gratitude to ABP for selecting me for this award. I thank to my institution, my dean, Dr. P. Shanmugasundaram sir, as well as Dr. Batmalada madam from Vijaya Institute of Principal Vijayawada, and my colleagues, friends, and students for the continuous support, encouragement, and trust. The guidance and motivation have shaped my journey and helped me to grow as a teacher as well as a researcher. So thanks once again, and also to share this special moment with me. So especially very much thankful to me. Thank you, sir.
Congratulations, Balaji.
Thank you, sir.
Our next awardee is for the Young Pharmacy Teacher Award 2025, Dr. Sunit Kumar Sahu, who is lecturer at UDPS, Utkal University, Bhubaneswar, Odisha. Congratulations, Dr. Sahu. You can share your views.
Thank you, sir. So, good afternoon to all respected dignitaries present over this platform. I'm truly honored and humbled to receive this prestigious award. My sincere thanks to APP and CEC for recognition of my profile. This award is not just a achievement, it reflects the support, encouragement, and inspiration I have received from my students, my colleagues, and my mentors. I would like to dedicate this recognition to all my students who have motivated me to improve, and to my institution for providing me the environment that nurtures teaching and research excellence. So again, thank you, APP and Professor Rajiv Dahiya sir for this honor. I look forward to continuing my journey with greater passion and contributing to our vibrant scientific community. Thank you, sir.
Congratulations, Dr. Sahu. Our next awardee is Young Scientist Award 2025, and which goes to Professor Deepak Kumar, who is Dean of School of Pharmaceutical Sciences, Sulini University, Solan, Himachal Pradesh, for his quality research potential. So, Professor Deepak, if you are with us, you can share your views. So, I think he's not there. We can move next. In student categories, we have from all over India, we have selected about 11 to 12 students for the APP Outstanding Student Award for their distinguished academic potential. They have supplied their academic merits, and on basis of that, those are selected. So first Outstanding Student Award goes to Miss Aditi Mehti. She is B.Pharm student of School of Medical and Allied Health Sciences, Brainware University, Kolkata, West Bengal. So congratulations, Miss Mehti. You can share your views if you are with us. Okay, I think she's not here. Our next awardee is Mr. Parasun Bera, who is B.Pharm student from School of Pharmacy, Neotia University, West Bengal. Congratulations, Mr. Bera. If you are with us, you can share your views.
Thank you, sir, for this great achievement by selecting me as the Outstanding Student Award. It is my great privilege to be a achiever of this award. It's not an achievement, it is just inspiration and motivation for my future perspectives in the pharmacy field. I want to dedicate my award to my college, that is the Newton University, and my dean sir, that is Professor S.C. Dinda sir, for his continuous guidance, support, and beyond the classroom and outside the classroom, his environment about the study and research projects. So I am also thankful to Dr. Rajiv Dharia sir and APP for such type of encouragement.
Okay. Congratulations, Mr. Bera. Keep it up. Our next awardee is Mr. Priyanshu Kumar, B.Pharm student from SNS College of Pharmacy, Motihari, Bihar. Congratulations, Mr. Priyanshu. If you are with us, you can share your views. Professor Sivli is there, who is the principal of college.
Yes.
I have seen. Yeah,
Yes,
you can
sir.
share your.
Good afternoon to all of you, especially APP convention. Myself, Dr. Shivli Numani, Professor and Principal, SNS College of Pharmacy, Motihari. Mr. Priyanshu Kumar is a good academic record. He has completed here his B Pharma with 8.24 CGPA till third year. He is continuing processing his final year till he has also qualified GPAT in 2025 with all India rank uh, under 3000. He is a hardworking and a curious student. I wish her success, his success, and especially thanks APP Association to select as such type of student and award them for and motivate them. Thank you, sir. Thank you very much.
Yes, thank you, Professor Namani. Next outstanding student award is Miss Pusti Choatia, who is B.Pharm student from Parul College of Pharmacy and Research, Parul University, Ahmedabad. Congratulations, Miss Pusti. If you are with us, you can share your views.
Yes, good afternoon to all the dignitaries and participants. I am truly honored to receive this Outstanding Student Award. As a student of Parul College of Pharmacy and Research, this is a significant milestone in my academic journey. I am truly grateful for APP for this honor and also grateful for my principal, Dr. Vandana Patel ma'am, and my faculties for guiding me throughout my academic journey. This award motivates me to continue striving for excellence in field of pharmacy. Thank you very much.
Congratulations, Ms. Pusti. Keep it up. Our next award is Mr. Soham Ghosh Dastidar, who is a M.Pharm pharmacy practice student at JSS College of Pharmacy, Mysore, Karnataka. Congratulations, Ms. Dastidar. If you are with us, you can share your views.
Yes, sir. Good evening to one and all, and I am greatly thankful for this award. And I am greatly thankful to our principal sir, Dr. Pramod Kumar sir, and my guide and HOD for continuous support and guiding me through my whole process of my masters. And I am thankful for this award, sir. Thank you, sir.
Okay, congratulations, Soham. Keep it up. Our next awardee is Mr. Gagan D. URS, who is a PharmD student at JSS College of Pharmacy, Mysore, Karnataka, and he is in PharmD category. I thank for nominating Mr. Gagan for this award. If he is with us, he can share his views. Gagan?
Yes, good evening, sir, and thank you for giving me this award, sir. I am extremely honorable, and I thank my HOD and all my professors, and also especially thank Mr. Ramesh, sir, who nominated me for this award. Thank you so much, sir. Thank you, everyone.
I also thanks Professor Ramesh for nominating the excellent students like you. Next award is Miss Archaya C, who is M.Pharm pharmaceutic student from JSS College of Pharmacy, Ooty, Tamil Nadu. Congratulations, Archaya. If you are with us, you can share your views.
Yes, sir.
Yeah. Thank you for giving me this award. And I would like to thank my mentor, Dr. N. Shahwa sir, for guiding me throughout this um, project. Thank you for everyone.
Dr. Jawahar, you are with us? No. Okay. Congratulations,
Yes,
Akshay.
yes, yes,
Oh, you are
yes,
here. Yeah. Yes.
yeah,
Some words for your
yeah,
student.
yeah. Thank you so much. Yeah, I thank APP for recognizing my student as the best student in the category of PG female. Thank you, one and all.
Okay. Thank you. Our next award goes to Miss Bhuvneshwari Mishra, who is M.Pharm Pharmacognosy student at Department of Farm Sciences, Utkal University, Odisha. Congratulations, Miss Mishra. You can share your views.
First of all, thank you so much for this honor. I'm truly grateful to receive this award. It means a lot to me, and I sincerely thank all my teachers, mentors, and everyone who support me throughout the journey. The recognition encourages me to work even harder and continue to improve myself. Thank you for giving me this opportunity and for believing in me. Thank you so much.
Okay. Congratulations, Miss Mishra. Next awardee is Mr. Rajat Singh Rathore, who is a PhD scholar at Patel College of Pharmacy, Madhyanchal Professional University, Bhopal. Congratulations, Mr. Rathore. You can share your views.
Yeah, sure, sir. First of all, I'm truly humbled to receive this award, and this moment would not have been possible without my mentor, Dr. Vita Maurya, and my institution. And I sincerely thanks to the organizers and specifically Dr. Rajiv and the jury for recognizing my work. This award inspires me for keep learning and growing. Thank you so much to everyone present here.
Keep it up, Mr. Rathore.
Thank you so much, sir.
Next awardee is Mr. Sanjay Chokalingam, who is a PharmD intern at Department of Pharmacy Practice, School of Pharmaceutical Sciences, Vistas, Chennai, Tamil Nadu. Congratulations, Mr. Sanjay. If you are with us, you can share your views. Okay. Our next awardee is Mr. Prince Soni, who is M.Pharm pharmaceutic student from Srimati S.M. Saha College, Pharmacy College, Mamdabad, Gujarat. Congratulations, Mr. Soni. If you are with us, you can share your views.
Yes, sir. Thank you so much. Good afternoon, everyone. Firstly, I am very thankful to the Association of Pharmacy Professionals, APP, for providing me this great opportunity and also be a part of this 14th annual international convention. And it is also honor to receive the APP Outstanding Student Award. I am truly grateful for this recognition. And it is totally possible because of my total guidance in BPharm and MPharm under the guidance of Dr. Hitesh Kataria, who has supported me a lot in this whole out journey. And thank you so much for this award.
Congratulations, Mr. Soni.
Thanks.
So one of our one awardee is in the best teacher category, and it goes to Professor Jyoti Basini, who is a professor of pharmacology at Seven Hills College of Pharmacy, Tirupati, Andhra Pradesh. Congratulations, Professor Basini. If you are with us, you are welcome to share your views. Okay, so finally we comes to our APP Memorial Award winners with outstanding research skills. So I want to see how many awardees have left or all are here with me. Okay. So I got the jury result. First award is late Srimati Khajani Devi Memorial Young Researcher Award 2025 with a check of rupees 10,000. And this award goes to Miss Joyita Tahseen Khan, who is a PhD scholar at Department of Pharmaceutical Sciences, College of Pharmacy, University of Arkansas for Medical Sciences, Little Rock, USA. So congratulations, Miss Khan. If you are with us, you can share your views. No. So second award, which is in the memory of Srimati Bhagwati Devi, and it is also a young researcher award. I'm taking little time because I have result, I have to match with the.
The request to keep the videos camera on because it's like it's good for records.
Yes. So second award, which is in memory of Srimati Bhagwati Devi, it goes to Miss Dhruvi Prajapati. She is assistant professor at Department of Quality Assurance, Parul University, Vadodara, as well as PhD scholar at KPGU, Vadodara, Gujarat. So congratulations, Miss Prajapati. If you are with us, you can share your views.
Thank you so much, sir, for this award. This means a lot to me. And thank you so much to my guide for providing me the guidance for my research.
Okay, congratulations, Ms. Dhruvi.
Thank you so much, sir.
Okay, next award is late Srimati Fulvasa Devi Dubey Memorial Award. It is also a category of Young Researcher Award. It goes to Miss Sarah Grace Andrew, who is a PhD student at Faculty of Pharmacy, AIMST University, Semeling, Malaysia. Congratulations, Miss Andrew. If you are with us, you can share your views.
Good evening, sir. Thank you so much once again. I'm really so surprised to be receiving a second award. Once again, I really like to thank my supervisor, Dr. Floria. Thank you so much, sir. Actually, it's truly a surprise to me. Thank you so much.
Okay, congratulations. Okay, last Young Researcher Award is again goes to a international community person. and he is none other than Mr. Mangesh Pardeep Kulkarni, who is a PhD scholar at Department of Pharmacy, Graduate School of Health, University of Technology, Sydney. Congratulations, Mangesh. If you are with us, you can share your views. No.
Thank you very much for this honor. I am truly grateful to receive this award. This platform is really remarkable. As I told earlier, I would really appreciate the rationale behind the support, and I would sincerely like to thank my mentor, Dr. Kamal Dua, and the association for this nomination. Thank you very much.
Thank you. Congratulations, Mr. Kulkarni.
Thank you very much, Professor Dia.
So this award was in the memory of Sri Bholanath Nikra and also in the Young Researcher Award category. So this completes our ceremony. We have upcoming APP conference. We are hosting APP Indo-Brazilian International Conference at Arya College of Pharmacy, Jaipur, Rajasthan in hybrid mode on March 14, 2026. And you are welcome to attend that. So I thank you all of you for your time. I congratulate all the awardees, and I would like to invite now Professor Teredina for the valedictory remarks. Professor Teredina. I think she left.
Check mic.
Professor
She's
Teredina.
there, but
Okay, sorry. Sorry, I thought I was already hearing and not. Thank you, Professor Kahanan, for advising me, and also Professor Rajiv. Well, in the name of you, Professor Rajiv Daya, I'd like to express my accomplishment to all the APP branches, professors, pharmaceutical professionals, students. You are wonderful. Greetings to all respect and relevant colleagues from India and around the world. Congratulations for organization, for presentations. It's a great opportunity to receive, to be among you, my friends in the pharmaceutical world. During my life, participating in several meetings and conventions in European countries, Asia, in Trinidad Tobago, in America, Caribbean, and all the world, pharmacists are recognized as one of the most important professionals. Our possibilities of studying reactive molecules, their safe administration to treat diseases, clinical trials, pharmaceutical care, safety of patients. We go from the lab to the bed of the patients. Delivery systems, people health, learning machine. It's a world of opportunities, and we could see during these presentations and in other presentations from APP. So in this moment, I like to valorize your work. APP is very valuable. Pharmacists work with patient, with heart. It's in the DNA of the pharmacist. Congratulations and thank you so much, Dr. Rajiv Daya and all the colleagues. we can see the branch new as Korea, and I'd like to make a mention about Vijay Institute of Pharmaceutical Science for Women, when we can read, knowledge is power. Really, knowledge is power. It's a very important statement to include that science today, medicine tomorrow, cure and recovery, comfort to the people and to our patients. Thank you so much, APP members, APP branch, and in particular, thank you very much and congratulations, Dr. Rajiv Daya. All you are in my heart. Thank you very much.
Thank you, Professor Terezina, for your kind words. So, before going to the hosting college, so I want to hear from my colleagues, starting from Dr. George. You want to share some views? Dr. Jensiana.
Yes, it was my first conference in this PhD, and I'm very happy, very proud, very proud to be with you. And I like very much, and I like to come to join your future events.
Okay, Professor Ghodake.
Yeah, thank you so much for organizing this award session and the presentations. I have no words. Actually, I'm sorry for recognizing the potential of those kind of the award ceremonies, but I feel that you have been doing from a long time, and I greatly honor you and your contribution to our Indian new young students, because that's a remarkable. And honoring their achievements is become like a catalyst, and those kind of catalysts will be always remembered in their lifetime. And that can be like we can bring one seed, like this award is like a seed for all of us, and to bring, inspire together, motivate together. And I would like to thank the leaders of the app and all branch presidents and vice presidents together. And I will promise you that the Korea branch will flourish at the greater extent you never imagined. And thank you for always motivating me and giving me the great insight about the like Vikasit Bharat, how we can join together hands and contribute actively. So your vision is much appreciated, admired, and I would see that all of our colleagues together, because this is international organization. So this organization is supported throughout the Indian states, as well as the globally recognized eminent scientists and the professors. And that's why together, suppose two people join hands, it becomes not 2 times, it becomes 10 times or more than that. We can't imagine that power together. And unity together bring the solutions for the challenging issues like multi-drug resistance, especially the pharmaceutical issues, cancer, and those can be recognized as historical contributions in our lifetime. So we will do very actively, join hands, collaborate closely, and bring extraordinary, impactful outcomes. Thanks for giving this honor to give me the vote of thanks, and appreciate all guest distributed dignitaries and all leaders of the app and all faculty members, students, and esteemed participants together, because those we can aspire, those young students, those was listening very patiently. I was looking for them. So these are our power, because we are going to give them all kind of support advices and make them leaders, global leaders. And that global leadership will be never ending. It's like eternal organization. It should continue generation after generation. And I again thank you, Rajuji. It's your efforts is not enough to be just admirable, but your honor to all of you, and it comes collectively back to you. And then together, imagine your power. This lifetime award is not just award or something, but is really catalyst to build their future career. I also thank Vijaya Institute of Pharmaceutical Sciences for Women Autonomous Vijayawada, and then SRK Foundation, the patron, or Aparao, and the Sri Krishna co-patron, and again the Kamal Doaji, Professor Kamal Doaji, and the Padmaltaji, Hemendraji Gautamji, Kuntal Dasji, Gaurav Guptaji. I will not describe, but the special thanks goes to the organizing chamber and the founding president of AP, uh, uh, the former director at School of Pharmacy, the University of the West Indies, Trinidad Tobago. So when he visited Korea, I will give you the example. I got inspired by his visit, and his visit was very tight, like 1-2 days gap after another country, USA, and then Korea, and a couple of other countries. And I saw this energy at this age. It can motivate our young ones to speed up or become a dynamic. And I am very depth on deepest respect for, or most respectful gratitude to the esteemed Professor Raju Dayaji. Keep inspiring, motivating us. We are wholeheartedly with you. Is you are the visionary leadership, commitment to the global scientific unity is like unwavering encouragement. Researchers across the globe, worldwide, including myself, are truly transformative because of this kind of events happening in your life. And receiving this kind of honors, I'm and recognizing my leadership is like great privilege and a great blessing. So I will just mention the few names. My PhD advisor, esteemed Professor Sanjay Govindarji, Jyoti Madam, who was also co-advisor and my inspiration that time, and she is now Vice Chancellor of the Shivaji University Kolhapur. And here, Danguk University leadership, the Vice President Jung Suk Sang ji and Dean Professor Deng Kim ji. Those are like Buddha-like figures for me. And my postdoc Professor, History Professor Deng Sangliji, who is the Kanmuk National University, I will reach out this organization to them as well. And let me give the first, because of the I am what I am because of my grandmother, who showed me the vision. Oh, you can be landlord, just believe. But nobody believed. So I get hurt, and I get to prove that in that journey, I become passionate about my education and the career together. And I thank my grandmother, grandfather together, and my close relatives, my mother, father, and my especially my wife. She's not living right now with her sacrifices, my kids' sacrifices. I have two kids, 6 years old, Sri Harsh and Sorangi. They are just like I'm feeling that God's blessing me because of my sacrifices and recognizing my leadership, like the esteemed Professor like Raju Dayaji. Thanks so much. And
Thank you.
one more thing, sir. One more thing. When I was undergraduate, the one professor was very close to me, like caring like baby, like caring for me. And then I was beloved student, one of the students from among them. They gave the best student award on the occasion of the student teacher day that time. So they've been out of that. I just mentioned my inspiration. First inspiration is the Amar Singh Bapu from the Srimant Babasaheb Deshmukh at Padi College. He's been my inspiration that time. His aura was like vibrant. And then the Professor Hasbes, Professor Lohar, Professor Jadhav, and Principal Karande sir also being like just like mentors that time, just early time. And finally, I say one sentence that I am very grateful for all these influential people in my life, and I just followed their path. And this academic and industry academia collaboration achievement is going to be take the next greater uh, outcomes and impacts in the greater global good. Like in case of the environment protection and controlling some of mitigating the multi-drug resistance and biodiversity losses. And climate resilient agriculture will be my priority to help our rural farmers and bring the rural development, women empowerment, and student empowerment together to bring a dream of India, our beloved Prime Minister Narendra Modi ji, who showed us to be a developed country by 2047. Let's join hand as a global leadership together and bring the glory to our India beloved country. Thank you so much.
Thank you, Professor Godaki. So we have some other persons here. Dr. Hanachi has joined from the Tunisia, so she can share her views, being now being as the secretary of the APP African International Branch. Dr. Hanachi. I think she left. She left a message for me. Okay. Professor Barik, you are here with us? Archaya, the student from Brainware University. Satish, you are with us? Dr. Bimula. Okay, I think all are in silent mode now. So, Professor Padmalata, you are with us?
Yeah, I'm available.
Oh, Satish is there. So, Satish, you can share your views.
Yeah, thank you, Dr. Rajiv Dahiya. So it's showing your great effort for organizing this kind of things, especially for awarding the students. So I heard that 11 students from India as well as 5 international students. So it will boost up their further career. So it's very nice thing for awarding the students. And also I'm thankful to all the international and state branches presidents and everyone. Thank you. Thank you very much.
Okay. So, Dr. Hanachi, you are there? Dr. Hanachi, I'm seeing you if you want to speak few words.
Yes, Dr. Shiv. Hello, everyone.
Yeah. You can put your video on if you want. Yes, hello.
Hello, everyone. Thank you for this opportunity. I'm very pleasure to join your association.
So your name was announced as Secretary of the APP African International Branch. Now we can start the activities. Okay.
Thank you for this opportunity.
Okay.
Thank you.
Okay. Thank you. Any other who want to share any views, or we can conclude here? Okay. We have anybody from the hosting college? Professor Padmalata? No? Okay. All left. So I conclude here. Thank you one and all for your support, for your time, and I will keep continue honoring student and talents among staff and students throughout the world. That is my well wish, that if any talent is there, let's extract it from the crowd and give some honor so that they can do their best in their life. Sometimes it does not matter what we do. We have done a lot of for ourselves, but is there is any possibility to do for others who, and or we can boost others to do great things. So that is my one of the motive I'm trying my best, and your all support is welcome in that initiative. So I thank you all for your time and making this event highly successful. Tomorrow will be the physical event, which the hosting college will see with the two speakers and their local award ceremony. So thank you. We
Thank you.
conclude here.
We thank you.
Thanks so much. Thank you.
Thank you very much, Professor Vidya.
Thank you. Thank you.
Bye.
धन्यवाद जी सर जी धन्यवाद सब गए
ठीक है सर, मिलते हैं.
हाँ जी बहुत बहुत
In
शुक्रिया
in March we have other engagement where we are going to honor you. Okay.
Sure.
Professor Teregina, in March 14 we have fixed one of the event keeping you as speaker. You will be online in that event. March 14, 2026. Your microphone.
Okay. In March 14.
March 14, yeah. It will be, you will be online. Yeah.
Yeah.
So one event is
Thank
fixed.
you.
I will send you the details when the brochure will be ready. Okay.
Thank you so much. And also, I'd like to do a proposal to all the colleagues, perhaps younger than I. I'd like to suggest a special remark, a special distinction to you, because
Thank you.
Dr. Rajiv gives recognize to all of us. I think that it's the moment, perhaps in the Korea or in another place, perhaps in India. I don't know where. I can't invite you in Brazil because I'm not in a good situation for this proposal. I'm very happy, but I think I am certain that Dr. Rajiv Daya deserves a special remark. Think about, please.
Okay, thank you.
Okay,
thank you.
Sir, I want to say few words. Can you allow me?
Okay.
The opportunity for all of our colleagues, and then I just missed to mention because of short of time that there is a big opportunity for the collaboration academician and industrialist because of we Dangote University have the big sum of the project that is called the RISE program that allows global talent to contribute the industry R&D solutions and the revenue model generation. So they are most welcome, and that's like rewarding program for those industry associate them. And then I think there's travel and all those issues will be no more. N number of colleagues can travel if they go through that program. So I welcome all of you and keep consider that program. I will maybe mention those other details because it's short of time. So how to proceed, what is the protocol and all those. Thank you so much, Rajuji, and all our team and all of our presenters and participants together. It's all going to be great leadership. We leadership bring to our young ones and make them hero in the world, global talent and exemplary. Because we know the experiences, we should translate them, not struggle because we lost many things, time and money and a person. We gain those insights. We should translate them. They should not struggle. They should be get some kind of the foundation stones, and then build their buildings whatever they can and have the global contributions, not only the India but global contribution together, and that global collaboration bring back to the India and under the our vision and the mission for to make our India glory to give back the depth of our soil and the nation we are going to build in the future in few decades. Yeah. Thank you so much.
Okay. Thank you all. We close here. Bye-bye.